Hello everyone, welcome back to Ask a Scientist Gaming, where we combine mediocre gameplay with expert science. I'm Ken Hansen, a professor in the Department of Chemistry and Biochemistry at Florida State University. I'm a photochemist slash photophysicist. I shine light on molecules and materials, and I want to know what happens to that light energy. We use spectroscopy to track that energy, and ultimately, hopefully do something useful with it, like making or breaking bonds, sensing things, making photocurrents, making mechanical work from photon energy. But more importantly, joining me today is Dr. Lisa Chadwick. Lisa, if you wouldn't mind introducing yourself.
Hi, I'm Lisa Chadwick. I'm the Deputy Director of the Division of Genome Sciences at the National Human Genome Research Institute, which is part of the National Institutes of Health. And I oversee our programs in rare disease genomics, like developing new methods to identify the genes and the mutations that cause rare genetic diseases. In our division, we also support a lot of research that's just basic foundational research and technology development in genomics.
Nice. Equally important, what game are we starting with?
Well, we're starting with one of my favorite games, which, to note, I have not played in probably 10 years, Super Paper Mario.
Let's do it.
Okay.
Let's dive right in.
See, I started a new save file, Lisa 2026.
It's a time capsule.
Yeah.
What I will note is Lisa actually brought over her WiiU.
I did.
And we plugged that in with the HDMI. So no emulators tonight, at least not till later. We're playing legit hardware right now.
Yeah, I have always been a gamer my whole life. And so I've had so many different game systems. And apparently I'm also a hoarder because I still have most of them. And my daughter just decided to get them all out and catalog everything and try to sell them. So that's how I remembered that I had a WiiU. I didn't even remember I had
them. No, usually it's the parents throwing out their kids' old crap, but you've gone the reverse route here.
The one with all the crap.
That's awesome.
Okay.
You kids just don't feel the nostalgia for these games that you did.
Well, my daughter has been playing the Wii games with me. Like, you know, they play like, what does she have? PlayStation, whatever number we're on.
Yeah,
5?
5 is the latest, yeah.
Oh. Okay.
That's fun.
Yeah, so she's into that. I am more of a Switch person now.
Nice. Switch or Switch 2?
Well, I do have the Switch too.
Okay.
Yeah. Okay.
Just checking. You're a legit gamer. You're in it.
Yeah.
It's awesome. But yeah, anyone that's not familiar with the WiiU, I think it was the first Nintendo that had like the handheld plus the console.
Yeah.
Plus the motion sensor controller and the nunchuck.
Okay.
So yeah, we'll show you guys that a little later, presumably. But yeah, if you want to give them context into what Paper Mario is, because this one had a very unique mechanic in the spectrum of Mario.
Well, and you can kind of see that. So you're flat, but you can also change the dimension of the game like that. And there are some things that you need to change the dimension of the game to be able to do. Like probably getting to whatever comes after this. And you only hit, though, that bar up there, you can only be in that other dimension for a certain amount of time.
Oh, interesting.
Okay, so
push that box. Cool Rex, welcome back. Where are you a professor at again? I'm a professor at Florida State University. So, yeah, officially, since the last time you were on the stream, I'm now a full professor.
Oh, well, so, congratulations.
Yeah, I've made it. Now I've just quit, right?
Yeah, that's how it works.
Yeah, exactly. Living the dream. And yeah, Lisa's a
not a professor
at NIH.
I am a scientific administrator now.
Who lives in Tallahassee.
I do.
Yeah. So yeah, Misadventures, welcome back. Thank you for joining us as usual. Yeah, if you guys have questions, feel free to put those in chat while Lisa dives in. Are you feeling it? It's coming back quickly.
Sure.
It's Mario.
Yeah, the thing about Mario is like you pretty much always know how to play Mario.
That's what's brilliant about it. And they were very intentional in how they designed the first few levels. Because it's always like a walkthrough of the mechanics.
Oh, geez. Okay, so I died, I think, because I went too long in the other dimension.
Cool Rex. Oh, that's great. Yeah, I'm actually a student at FSU. Well, that's awesome.
That is awesome.
Which means you're starting classes in like 5 days. It's Mondays when they start. So yeah, Cool Rex, I'm both happy and sad for you. But thank you for joining us to hang out the Wednesday night before classes are back. What a great time to have a drink in hand, playing video games, talking about science, talking about all sorts of topics.
Oh my gosh, there has to be a better way to do this. Like, maybe I have to do this.
Oh, yeah.
Yeah, that's what I have to do.
That's fun. Like it. So I have no idea. I've never looked it up. What's your guess on the speed run for Super Paper Mario? What is the fastest someone has beaten this game?
The thing I remember from playing this game or doing this stream with you before is that it's always 8 gajillion times faster than I could even imagine.
Yep.
And so I'm gonna say, I don't know, like 25 minutes.
What's crazy is this one is 3 hours and 44 minutes. This is one of the unique exceptions to that. I'm guessing there's a lot of cutscenes and there's not a lot of ways to break the game.
There's a man
So yeah,
here.
3 hours and 44 minutes. Somebody committed to beating this as fast as they can.
That's real. I mean, that's an endurance sport.
No, that's exceptionally
basically.
long. Like, I think Halo's under 2 hours. It's crazy that it takes this long.
I don't think I can do anything for
But yeah,
over 3 hours at a time.
I mean, what's crazy is, I mean, second place is only, 3 minutes behind. So it's not that ridiculously optimized, but yeah, that's fun. So next time you're on.
Yeah, I'll work on that.
I'm curious what that video looks like. It's probably very slow and janky.
Wait, okay, I know, sir. Okay. Yeah, I remember really, really liking this game. It is very different in that you can flip dimensionality.
And they brought that back in like Mario Odyssey, I think. It was one of the more recent ones.
Yeah, and I
Somebody
never
that
played any of the other. I think that there were other Paper Mario games too that I did not play.
Yeah, I think in Odyssey they had mechanics where you'd like go in a tube and then you'd be in 2D and then you'd go back. Not like a flip like this, but...
Okay, there's a lot of people talking to me though.
Yeah, I'll wait for that to be done before asking your first question.
Okay.
We need this important lore about Paper World. You answered poorly.
I guess I should have guessed. All right, well.
No worries.
Sorry, but I wonder if I have to go back and like get him to. I'm gonna try that again. I'm gonna answer red this time and see what happens. Maybe this doesn't matter.
Anyone in chat that's an expert at Super Paper Mario, feel free to chime in. We are not game purists. If you have feedback, we're happy to take it.
Okay. Oh, he's gonna ask me again. Okay. It's red. I should have guessed. I mean, the whole house is red. He's wearing red. Everything's red.
So many regrets.
Okay. Great. Yeah. I did need to do that.
It's fun. I really like the aesthetic of this game. It's a nice departure over the classics. But now that you've settled in and you're completely used to playing again,
right.
We'll start with the opening question. So you've been on the stream before, but it's been 4 years. 4 years, it feels like a lifetime. Many things have happened in 4 years to all of us. Yes. But for you, you started out as 6-year-old Lisa playing video games, thinking about what you want to do with your future. Presumably the thought was not, I'm going to be a deputy...
A government bureaucrat?
Yes. I mean, you said it, I didn't. But yeah, a government bureaucrat working on genomics and genetics and working with people on funding and all sorts of stuff. Living in Tallahassee, working out of a federal building. And so there were many twists and turns in that journey, obviously.
Yes.
So let's start with 6-year-old Lisa and the journey to get to where she is now. Peak achievement doing Ask a Scientist gaming, playing Super Paper Mario.
Exactly. Yeah, 6-year-old Lisa would be really surprised at where 50-year-old Lisa is now, because this is definitely not every little girl's dream to be a government bureaucrat. But I did always like science. I knew I liked science. I thought I wanted to go to med school. I think like a lot of people who like science.
Yep.
I don't really need to talk to you, sir. I don't know what we're talking about here. I went to a really cool magnet school for middle school that had a great science teacher who did some really cool projects with the classes that got me very interested in science. One of the projects we did, he basically created this whole murder mystery, and we all were trapped on an island of some like crazy wealthy professor who had developed a clone of himself, and then somebody killed the clone, and we had to do some detective work to figure out who had killed it.
Yeah.
In that class, is when I first learned about genetics, and we first started doing like Punnett squares and all that kind of stuff.
I just want to point out how progressive that activity was.
I feel like this
in
guy
terms of
just
pedagogy.
Oh yeah, that teacher, I mean, he really did a lot of really cool stuff.
Yeah.
What is that? Another thing that we did. Okay, so on the first day of, who's Old Man What's It? See, this game is a lot more complicated than I remember. You could see on the ceiling of our classroom this like black wooden box. It was there all year. Nobody knew what it was. And one day we turned up at class. We all had to go sit out in the hallway in 2 rows like we were on a bus. And we were taken to sort of a secret like lab where we were going to investigate a spaceship that had come down. And so we go into the classroom, and out of this box comes like an electric walk, basically with Christmas lights on it.
Yeah.
Which was a spaceship that came out of the sky, and a frog was in it wearing a spacesuit. And that is how we were introduced to the unit where we dissected frogs. And we had to figure out why this frog had come to Earth. And so he was a really great teacher. But anyway,
that's awesome.
That's when I got interested. That's when I really, really got interested in science, and especially in genetics. And then I went to college. I was a biology major. And I realized like towards the end of undergrad that I was probably not going to get into med school without some serious work that I didn't have time to do anymore. And I didn't know what to do. I moved back home. I worked at the mall for a while.
Oh, we need to know which specific store.
I worked at Express.
Nice.
I remember interviewing for a manager position.
Little did you know how much that would serve you in your customer service role as an administrator. It's a life experience worth value.
Yeah, so I obviously I didn't stay working at Express. I kept interviewing for jobs, and I ended up getting a job at, no, I don't want to talk to you. I just want to know how to get to whatever I'm supposed to do next. I ended up getting a job in a research lab at Harvard Medical School. It was a lab that was trying to identify the gene that caused one particular rare genetic disorder. And so I worked as a technician there for 2 years. I did
That's
all.
crazy. They hired externally, right? Rather than a student, or is that a normal?
I think technicians can often be hired external. And I worked there for 2 years, and then I decided to go to grad school. And I went to grad school, did a PhD in genetics. I really planned to be an academic too. That was my goal. I did a postdoc. The NIH has what's called an intramural side and an extramural side. So the intramural side is the labs that are at the NIH. And the extramural is where I am now, which is like the part of the NIH that gives grants to researchers in the community. And so I did my postdoc in an intramural lab. Now, I don't understand. I just came from there, didn't I? Why am I going back in? And my postdoc was great in a lot of ways. My postdoc advisor was really great.
Star on the wall? What is that?
Yeah, what is that? Great question. I feel like it's significant, and I don't know why. Because nothing is happening here.
Because that was a lot of work to get nothing.
I feel like I should have picked a game that's less complicated than this.
While talking, yes. No, it's perfect.
I didn't remember that it was this complicated.
If anyone in chat has feedback. But anyway,
Okay.
So after PhD.
So I did this postdoc, and as you know, did you do a postdoc
I did.
too? Okay.
Yeah.
It's very common for researchers to do a postdoc before they get like a real job. And you have to really nail it in your postdoc, right? You've got to be at the top of your game to be competitive enough.
Publish quickly, and yeah,
Right.
There's a lot of luck involved.
Exactly.
Yeah.
And so it happened that when I was doing my postdoc, I also had a couple of things happen in my personal life that really did not keep me on my game. And so I just kind of lost all steam and motivation. And I realized that I just did not have it in me at that time to do an academic job search. So then I was like, well, now what do I do? Because you don't know. I don't know in chemistry whether you get a lot of experience or exposure to other career paths in science besides being an academic. But you didn't in genetics for sure, and in a lot of molecular biology kind of science. And so I had no idea like what else I was gonna do. Now, did they just tell me to do something? Okay. And I applied for all kinds of different jobs. But one nice thing about being at the NIH is that I got access to new jobs when they were posted. And so when I was having this sort of like career crisis, this guy has to do something for me. I don't know what it is. I stopped. I don't have a pixel shaped like a hand. Oh my God. Okay. This is very complicated.
Yeah, no, it's hard. And you're reading while talking.
Really regret this.
It's fine.
Okay.
We can change at any point, but I mean, have a little fun with it, right?
Oh, that's the thing I need. Oh no, that's a star. But this guy won't let me do anything. That guy back there wouldn't let me do anything until I had made this guy happy. Wonder if maybe that up there seems like that could be important.
Yeah.
But how do I get up there? Not that way. This is the longest way to say. So then I ended up becoming a program officer. This job was posted. I had no idea at all what this job was, but my postdoc advisor did, and he encouraged me to apply for it. And they just happened to be looking for somebody with similar expertise to what I had.
Nice.
And so that was 18 years ago that I became a program officer.
Yeah.
And I've not worked in the lab since then. I've not done experiments since then. And I wish I could say I missed it, but I really don't.
That is fair. And recently, you were promoted
I was promoted about at the end of 2024. Yeah, so now I'm more involved in sort of the larger scale leadership of the division. You know, managing the programs that we maybe this is where I'm supposed to. I didn't go in there, did I?
Secret pipe.
Okay, please let this be something that helps me. That's not, this is just coins. This is not helpful. Yeah, so that's great. I also like, you know, being in a position where I'm able to support my colleagues more. That's something I've always enjoyed doing. So, yeah.
Well, and you get to see fun, cutting-edge science
And I do
keep
get
in the
to
loop on all that.
Exactly. Yeah, when you're a program officer, you see lots of different areas of science at a very at a very like broad and high level perspective, which is very different from when you are working in a lab.
Yep, you get tunnel vision.
Oh yeah, but it's interesting. I've learned a lot of things, and I like it.
Very cool. So speaking of jobs, so you work remotely.
Yes.
Post-COVID.
I do.
They shook your world and said you have to come back to the office.
Yeah.
And you did what?
Okay, so yes. So I have worked remotely. I need to be somewhere where I can actually do something. I have no idea what to do right now. We moved here in 2009, so a long time ago. And I was probably the first person. I'm gonna go like to an earlier part of this game.
Sounds good.
Is there I'm just gonna like, I just cannot. Now I don't even know how to get out of this. Oh my gosh. I've made a terrible mistake here.
Got it.
I need to go back to the beginning because I have no idea what I'm doing here. And I will literally not be able to talk until I figure out. Yeah, so I was probably one of the first NIH people to be remote. Not the first, but there were very few of us at that time. And it was a time where like I just called in to every meeting like on a phone.
Oh, yeah.
And I just listened to words coming out of the phone. In the room, I was just like this like disembodied voice. It was a very different time for working remotely. But I've done it for that whole time. And when the new administration changed, they eliminated all remote work. And so all of the remote staff had to figure something else out. And we didn't know for a long time what that was going to look like, whether we were going to have to move to these offices. Now, when I first moved here, I was based at the institute that's in North Carolina, but I don't work there anymore. I work for one of the institutes in Bethesda. So I was like, oh, great, I'm going to have to like move to an office that I've never even been to return. And I ended up, I'm just going to do a new game.
Let's do it.
And we're just going to call this. And yeah, so we've ended up spending the last year and a half trying to find other federal offices that will host our staff. There's actually at the NIH, at the time that this happened, I want to say that there was about 1,500 remote staff across the NIH.
Jeez.
There are fewer now because we lost a lot of staff through a whole bunch of different sort of staff reduction things. But still, we have hundreds and hundreds of remote staff. So we've been working with other federal agencies across the country to have some sort of partnership, and they have been hosting our staff. We've been hosting staff from other agencies. So I had been looking for a long time in Tallahassee. I personally had driven to like every federal office in town and asked if they had space, and nobody did. But finally, I ended up getting placed at the National Weather Service office, which you may not have known is on the campus of Florida State University in the Love Building.
That's awesome.
And so that's where I've been working for almost a year. It'll be a year in September.
Is that the one with the Doppler on top of it?
Well, I think that's actually on the other part of the Love Building or the newer building, the EOAS.
Yeah, the Earth, Wind, and Fire building. Yes.
But I have been to the Doppler radar that's at the airport.
Oh, that's fun.
The nice thing about working in the weather office is, I've learned a lot about the weather.
I believe it. You got insights into hurricanes and when you should panic. And
yeah. Do you remember when we had like tornado warning?
Mm-hmm.
Yeah, I was in the office that day.
And they're so excited.
It was, yes.
Do they have a situation room with a bunch of monitors?
Well, that's just what their office is.
Yeah.
It's just a big open room with a lot of monitors. When I first walked in there, I was like, I don't have enough monitors to fit in here. I only have 2. They have like 5.
Now you have 5?
No, I don't. I could not function with 5. I can barely function with 2. There's way too many windows open.
But they accepted you nonetheless.
Yes. So yeah. So it's. I think we all can agree that it's ridiculous, but it's been great. And then we also have people. I manage other remote staff. One of them works at an IRS office. One of them works at...
Much less exciting than the Weather Institute.
It is. He has a panic button on his desk, though. I don't have that.
Interesting.
One of them works at a Forest Service office. We've got people at Department of State warehouses. It's really everywhere. And I would say what I really liked about it was how cooperative everybody was. All of these agencies had remote staff that they were trying to find homes for too. And everybody really sort of came together and was very cooperative. And that's not something that you see every day. So
that's awesome.
Pretty interesting.
Everyone came together.
Yeah.
Suffering through the same challenge. Really appreciate that. All right, Cool Rex has a question. Before you answer, I'm going to pop up one of our predictions because why not? It's related to the question. Hi, Lisa. Can you elaborate on your experience on how you've gotten into rare diseases? I currently have a client that's in the therapeutic space looking to find cures for rare diseases, including Rett syndrome.
Oh.
So I really wanted to learn more about rare genetic mutation space, especially when I'm speaking to some of these scientists, etc. And so I'm going to pop up a question for you guys. Lisa, Ask a Scientist Gaming, we ask you guys questions via the prediction mechanism on Twitch. And so Lisa's written a bunch of these questions. We're going to pop one up right now that's related to rare genetic diseases.
Yep.
And so I had to shorten it to 45 characters, whatever it is. But the actual question is, what percentage of people that are thought to have a rare genetic disease will get a diagnosis after genetic testing? And so this is, what's the true positive rate of testing? Is that basically what it's asking?
Well, probably not that, but the diagnostic rate.
Yeah.
That's what we call it.
Of after genetic testing. And
Yeah.
this is just like a standard genetic test.
Yeah, we can talk too about like how they do genetic testing.
Okay. So what percentage of people that are thought to have rare genetic diseases will get diagnosed after genetic testing? Is the answer 70% or 40%? And those of you not familiar with Twitch, all you have to do is click that predict button at the top of your chat right now, and it'll give you 2 options, and you can bet on either of those options. What's nice about Twitch imaginary internet points is you can bet as many internet points as you want, depending on your degree of confidence. And so, yeah, also, if you're not following us, click the follow button. You'll get more imaginary internet points. Useless outside of the stream, but on the stream, you can make us take a drink. You can unlock an emote. You can highlight your message. You can, I don't remember what the other things are. But yeah, enjoy your internet points. And note you're on Ask a Scientist Gaming Honor Policy. You can't look this up on ChatGPT or Google or whatever it might be. Do it based on what you know and what you've seen of Lisa's gameplay in Super Paper Mario.
I hope that you're not getting anything from my gameplay because I haven't managed to even get past 1 level. So.
But they're brothers, so there's genetics involved here. So
right.
You guys learned something from Mario.
One's short, one's tall.
Exactly. We've all seen the movie Twins. Okay, we have. We're old enough to have seen Twins, but maybe not the younger generation. Yeah. All right. I'm going to copy paste this. All right, you guys have about 10 seconds left. Put your answers in there.
Okay, there's so much lore in this game. We have to
All
go
right,
through.
Lisa, what is the answer? 70% or 40%?
So it's actually 40%, and that's probably a pretty generous estimate. It's really interesting to me because, like you said, when I started my career, I started in rare disease genetics. That was, almost 30 years ago. And it is crazy to me to think about how much has changed in that time period. It used to take, if you had a rare genetic disease, it used to take years, literal years to figure out even what the gene was that caused it. And that's really like that you had to figure out what chromosome it was on. This was before the Human Genome Project. You had to find all of the genes in the region of the chromosome where you thought your mutation might be, and you had to figure out what they were, and then you had to figure out how to sequence them and find mutations. It took forever. They do it very differently right now, and it's very fast. Like the sequencing part of it is fast, but actually the chance that you're going to figure out what the correct mutation is that causes the disease is pretty low. So I actually lead a research consortium, which is called GREGOR. GREGOR stands for Genomics Research to Elucidate the Genetics of Rare Disease, which is very exciting. Acronym
Gregor
that I did not come up with. But one of the things that we're trying to do in that program is figure out ways to identify the causal variants in rare genetic diseases that are going to give more people an answer. So one thing to know about how we do genetic testing right now is there's a couple different ways. One of the main ways is called whole exome sequencing. So basically what that does is that sequences the protein coding parts of your genome, the exons, and those are where it's most likely that you would have a mutation that would disrupt the function of the protein in a way that would give you a disease, right? And so you take the people with the disease, you look for mutations in those regions, you look for things that are found only in the person with the disease and not in people that don't have the disease. And then you're like, great, here's the causal variant. But it's actually so much harder than that. So one limitation of exome sequencing is that it is only looking at that very small portion of your genome. And your genome is much bigger than that. And there are other parts of it that do things that we don't even fully understand yet, that could also contribute to you having a disease. There's also different kinds of mutations that could happen that are not detectable using whole exome sequencing. So they're not very good at detecting mutations that cause, for example, defects in the way that the exons are spliced together. There is not a good way for exome sequencing to identify structural variants or like larger variants where there's like a duplication or some other sort of larger than one SNP or one mutation kind of variants. And so we've been interested in figuring out ways to look at more of the genome with other technologies, to see if we can figure out how to identify causal variants that are not picked up by this sort of standard method of genetic testing.
I mean, given the challenge of it, 40% doesn't seem that bad, actually. I mean, is that a reasonable rate?
I mean, it's great for those 40% of people.
Yeah.
But it's not great for the remaining, 60 to 70% of people.
Yeah.
Who go sometimes for years trying to find a diagnosis. Now, what am I supposed to do right now? This is the problem. He's talking to me and he's telling me what I'm supposed to do. Oh, okay, great. Probably have to talk to you. Yeah, and so it can actually take years for somebody to get a diagnosis. That whole time they're going from doctor to doctor. They're waiting for therapeutics. They're waiting for other kinds of support, and they don't know what is wrong with them, right? And so we want to shorten that time period as much as we can for people.
And so going back to Cool Rex's question originally is, can you elaborate on your experience and how you got into rare diseases? And it sounds like you accidentally got hired into it and found you liked it.
Yeah, I mean, I liked genetics, but I just happened to get hired into a lab like that. And then actually, I left that for a long time, so I didn't do my PhD on that. I did my PhD on X chromosome inactivation. That's related, actually, to Rett syndrome. Rett syndrome is caused by a gene that is on the X chromosome, MECP2. And MECP2 is a DNA methylation binding protein. And so it's involved in epigenetic regulation, which is the way that cells turn genes on and off.
That's very fun. All right, cool, Rex. I hope that answers your question. I don't know if that helps you learn more about genetic mutation space, especially when I'm speaking with some of these scientists. But yeah, it's a complex problem, but you're working to fix it, which is awesome. So check out that Greger Institute. I put the link in there in chat if you guys want to take a look. The backronym to get to Greger is pretty impressive. All right, so oh man, before we dive into the can of worms, so we were talking earlier about your experience with your intermediate school teacher, doing all these amazing labs. There was a Reddit post recently about, like, there used to be a lot of like genetics related testing you could do in these, like, you know, tabletop kits, but there were more sometimes where students would discover things about themselves, like brothers and sisters maybe not aligning, like, you know, these like poly X or all these different things that can happen.
Yes.
And so people are shying away from that more, apparently. Have you heard anything about this?
Well, I mean, it is true that when you start to look into your genetics, you may learn things that you didn't want to know. One of them could be about your parents. That's one possibility.
Parents in quotes.
I remember when I worked. Now, am I supposed to be talking to you? No, you're not the person. I remember when I worked in the lab as a technician. We had this like big cohort of individuals with a specific disease. We had like a whole pedigree, and I was doing the genotyping for those individuals. And you would definitely find cases where the family had told you who was related to who and how, and the genetics did not agree with that. And so that is one thing. Also, you can find out that you, sure, I'll save my progress. Why not? You can find out, even with genetic testing, things that you may not be prepared to hear, right?
Yeah.
If you, for example, if you have a parent that has well, where is he? If you have a parent that has cancer, for example, and you get genetic testing for cancer, we have people called genetic counselors who understand that and who can help you sort of hear that information, help you understand what it means, and help you think through like what your next steps would be.
Because even then, it's still a probabilistic outcome, right? It's not guaranteed.
Right. Yes. You hear people say, oh, I have the gene for whatever.
Yeah.
Like, no, we all have the gene. We are all human. Exactly. We all have that gene.
Did you express it or not?
Right. But you have a mutation that makes you more likely to develop the disease that's associated with that gene. It's not even in a lot of these rare diseases that we work on in Greger are thought to be caused by a mutation in just one gene. But even in those, it's not necessarily that if you have the mutation that you're for sure going to have the disease. We know that there are other mutations that might modify your risk for that. Genes code for proteins, and proteins don't like work by themselves, right? Proteins work with other proteins. And so yeah, so there, it's never as simple as just one gene, but a genetic counselor can help you understand what your genetic testing result means and what you might want to do next.
I mean, what's crazy about that is like we're experts in science, and I remember this specifically when we like had our kids, where you have the DNA test, right? You test for Downs and all these other things, right?
You probably had the non-invasive prenatal testing,
Yes,
right?
exactly,
So
exactly.
Cool.
The blood test, yeah.
That didn't exist when I had children.
Oh no. but it was crazy meeting with the guy, because he was just like spouting off all these conditions, and here's what it is, here's what it isn't. And I'm a smart guy, but I'm not an expert in that domain, and I was lost. And you have the average person that never taken a biology class in their life, and they're trying to cope with this like single point mutation math.
Right.
That's non-trivial.
Right. So, so that non-invasive prenatal testing is a good example of a time where you might get a result that you are not prepared for, right? So not only might you get a result you're not prepared for about your child, you might get a result you're not prepared for about yourself.
Ooh, they don't prepare you for that.
No. So the way that testing works in a very broad sense is basically that it's assessing the amount of copies of a particular DNA sequence or chromosomal region or whatever. And it's looking for sort of an even amount of copies. So the baby should have 2 copies of it. The parent should have 2 copies of it. If the baby has a trisomy, and so they have 3 copies of one of those chromosomes, you're going to see sort of a departure in the rate, I don't know what you'd call that. Like you're going to see more DNA, more quantitative DNA. It's basically looking for a quantitative amount of DNA of different parts of the genome. And so you're going to see that they've got too much of chromosome 21. Well, sometimes people would get a result where it said your baby has, a lot of different chromosomes that are duplicated. And it turns out that some of those times, what it's actually detecting is that the mother has cancer. Because that's also associated with chromosomal duplication and rearrangement. And those sequences are also found in your blood. And so what you're basically doing is you're looking in the blood, the mom's blood, which has sort of fetal DNA in it. But if the mom has a tumor that is causing, chromosomal duplications, you're also going to see that in the blood. And that's going to mess up sort of that quantitation of chromosome numbers. So I think that there are actually now like cancer screening tests that are based on the same sort of technology.
That makes sense. But man, what horrible news to hear, right? Because you get the gender of your child, but also you have cancer, but you can't treat it until after birth. Like, oh my God, or you decide otherwise.
Yeah, so actually at my institute, we have every year like an institute-wide sort of gathering and research sort of symposium. And we always have what we call a patient-provider conversation. And somebody who has had some sort of either genetic testing or who has been coming in for care for a genetic disease with one of our clinicians will talk about sort of their path to being diagnosed and what it was like and all these things. And we did have somebody come in one time who did find out that she had cancer when she was pregnant through her non-invasive prenatal testing. And she talked about how difficult it was to make those decisions about what to do about treating her cancer versus waiting for her baby to be born so that it wouldn't be impacted by the chemotherapy and everything. It's really quite interesting.
And that's intentional to like help you guys not lose sight of the patient-focused goals of this? Is that why?
Yeah, it is really it's interesting. A lot of my institute is a lot more clinical focused. I would say the people who work in the intramural side, they do a lot more patient clinical focused genetics. But we also fund so much that is just very basic sort of technology development, and you don't get to experience that. You know kind of what people are doing with those technologies, but you don't actually like. This is the most boring game. I think we should play something else.
What do you want to do?
Like, we really haven't even gotten to the level yet.
It's just walking through lore and backstory.
I've made a big mistake here.
It's up to you. If you want to change to something else, we can go either way.
Actually, there is like a different Mario on here that's not quite as like
intensive.
Yeah.
All right.
Let's do that.
Transition that. All right.
You've been sticking through watching me.
Oh, I mean, that's
play this like.
I'm here for the conversation. let's,
and it's hard to have a conversation because.
No, you're doing great.
Okay. All right. We're gonna try to play the other Mario game that's on my WiiU. I actually don't know which one it is. We're gonna find out.
Might be.
Super Mario 3D. Is that a game?
I don't know if that one was on the WiiU. I don't know. We're gonna find out though. That's exciting. All right.
Okay. How do we get to that? We have to go to here.
And it's downloaded on here?
Yeah. I did test that.
Nice. Okay.
The problem with it is you have to play it with this thing, which I don't actually love this thing, but
it's pretty clunky. It's not quite like the Switch.
We're gonna do. No, it's not. No, it's not.
All right. Well, that guy is loading. Equace. I don't know. How would you pronounce that? Equiace. Equiace?
Yeah, sure.
It has to be rough working for the NIH while it's been gutted, and a new director started hampering vital research around the country.
Yeah.
Short answer is yes.
I mean, it's definitely okay. Hang on. Before I can answer that, I have to figure out how to get to the game. Now, I feel like... Why is it not, like, moving?
You should project on the screen, right?
Yeah. I have to get down to that. But why is it not... This is the problem with having not played this video game system in a long time. Is it, like, not communicating with that? Maybe that's the problem. Maybe I've made a huge mistake just bringing this WiiU.
No, we got to do a little super.
I feel like that is what's happening. Okay. Okay, I got it.
We got two screens. Oh
yeah,
3D world. There we go.
Yeah. Okay.
Nice.
Sorry, everyone.
All right, we're good.
Yes. So it's definitely.
Well, so let's recount a timeline of events and how that has affected your life.
Okay. So, there's been a lot of things that have happened over the last year that you've all heard about in the news. If you read Reddit, you've heard about them. You know, whatever. So at the NIH in particular, we had a lot of staff losses. So you've heard about that in the news, I'm sure, that there were a whole bunch of different ways that government agencies lost staff. There was a time where we were getting emails, periodically from Doge telling us basically that we were not good at our jobs and we should just quit and do
retire?
something that was more, a better job, right?
I hope you saved all these emails. Like it's a time capsule.
It really is. So that was fun. We got those. part of that was that they gave a lot of like different incentives to people to leave. They gave you those emails that I was talking about where they were saying basically you should go and get another job and not work here anymore. We call those the fork emails, right? Because the subject line was like fork in the road or something. And basically,
I would have gone with fork you, but
right. So basically, they offered incentives to people to quit at that time, and so you could actually stop working and get paid and not do your job for the rest of, I can't remember exactly the timeline, but it was like 9 months, right? They were going to pay you,
yeah.
but you would not be actually working.
So if you're close to retirement.
Yeah. So
pretty
actually
sweet.
at my work, we didn't lose a lot of people to that. Very, very few people. I would say that in general, a lot of people were pretty skeptical about that because that's in the federal government, we do actually have like established ways to reduce staff. It's called a RIF, right? And there are rules to that. There's like a process to it. But this was not that, right? And so people were, I think rightfully so, not convinced that this was actually going to happen, right? And so we at my institute we hardly lost anyone that way. But they also used some of the actual like legitimate ways to reduce force.
I see.
And those are things like providing early retirement incentives. So people who had a certain amount of years of service or were a certain age or both, it was like a combination of things, could be eligible for that. Or if you were close to retirement, you could take a retirement, you could do your retirement and then you could also get like an additional amount of money as sort of an incentive to retire. So we lost people that way at the NIH in sort of these extramural positions where we make grant awards. We have all different kinds of staff. We have what are people like me who are called program staff. We're the ones who manage the scientific parts of the research programs. And then we also have what are called grants management staff. They're the ones that actually like make the grant awards. They deal with all the policies. They make sure that you're in compliance with everything. They do the money part of it and the rules part of it. And I don't do that. Thank God. I'd be really bad at it.
That's too administrative.
Yeah. So we lost a lot of those people, specifically at my institute. Other institutes lost staff in different ways, but that's just how it affected us. And that made it hard to do our job because we didn't have the staff that we needed to make all the grant awards that we normally would. They also did some more targeted reductions where they decide ZL. Okay, Z is that one. They decided that they were going to change the way that parts of the NIH were organized, and so they were going to consolidate different functions into sort of more central things, and that included things like purchasing and human resources and things like that.
And that was done without consultation of people privy to this structure, or?
I mean, who knows?
That's a fair answer.
You really don't know how they decided to do the things that they
Somebody saw a flow chart. Maybe.
So that is another way that we lost staff, and and this happened to all agencies. I don't want to make it seem like this is just the NIH. All agencies went through this. And when you lose staff in ways that you didn't plan intentionally, in sort of a random way,
and very quickly,
it makes it really difficult to do the function of your agency, right? And so that was kind of a tough year. We also lost a lot of NIH institute directors. So the NIH is made up of 27 different institutes and centers. Mine's the Human Genome one. But they all are headed by a director. It's usually, that person is a scientist who is a leader in their field, and they sort of set the strategic vision for the institute. They are sort of the final deciding factor in what gets funded and what programs go forward, etc. Our institute director was one of the first ones to not have his contract renewed. We had a very popular institute director. We used to have a really popular social media. Our institute director was not only a good scientist, but he was also very funny and willing to do basically anything. And so we didn't have an institute director for a while. That also makes it hard to function because you don't have anybody in a permanent position who can make decisions. So we did have somebody come in to be our acting director. It was somebody actually who had previously been at our institute. She was my division director, but she had since left and gone to another position elsewhere at the NIH. So that was disruptive. And then we did just actually get a new institute director announced. She is a scientist that we already knew. She's somebody on our council. I don't know how much you know about the NIH funding process, but like there's all these different committees basically that we get feedback from about grants that we're planning to fund. One is review. We get feedback from reviewers about the grant proposals, and that helps us identify the ones that are the strongest applications. And then we sort of make recommendations for funding, and we take those to our council. The council reviews that as well, and ensures that review happened in the correct way. And they sort of basically concur with the fact that review was good and that we can go ahead and fund applications that we want.
Nice.
So anyway, this woman was on our council, so she's definitely like a leader in the field, and we're very lucky to have her, and she starts actually on Monday.
That's exciting. I mean, so if you had to, I've heard the NSF is down 50% of their staff or something along those lines.
Yeah.
Where are you guys on that spectrum of?
So we're not as bad as that. I can't remember at my institute how many staff we lost. I do know the answer to that somehow, but I forgot. It was a lot, but it wasn't 50%. And it was in very specific kinds of areas, right? And so NSF, I think, lost staff much more across the board. My guess is that they also lost a lot of their scientific staff, like me.
Yeah.
They had a lot of other issues. They were moved out of their headquarters building.
Yeah, that's crazy. It's a new building too.
Yeah, which is I don't know a lot a lot about it, but I think it was purpose built for them. I think it had special special capability to do grant reviews.
Yeah. So who occupies that building now?
I can't remember, but it's not NSF. But I can't remember who it was.
Yeah.
It was somebody who I don't remember.
Yeah.
Not who is. Yeah, I don't remember.
No worries.
And I don't remember why they wanted that building, but
for the review
I think because it was new.
Obviously.
Yeah, right, exactly. They were gonna
got
do
pretty windows and yeah.
Yeah, I think it was new.
Good times. Oof. All right, cool Rex. Another question. Question is, when referring to funding at your job, are these funding for companies looking to advance clinical trials or pre-IND phase?
Oh.
I'm assuming if it's government, it's most likely grants and not equity investments.
Yeah, it's grants. Even so, most of what I'm talking about is grants that we're giving to researchers at universities like you. You're not funded by the NIH,
Nope.
but it's researchers at universities who are doing sort of the foundational research in these areas. We do also fund small businesses. There's the small business
SBIR.
SBIR.
Right?
Yeah, what does that stand for? Small Business
Business
Innovation.
I don't remember.
Uh,
There's like SBIR and STTR.
STTR.
Yeah. What does SBIR stand
Small
for?
business. What's probably innovation.
Small Business Innovation Research Program.
Okay. And what's STTR? Small
Small
Technology
Business Technology Transfer. Yep.
The main distinction, I think, between SBIR and STTR is the amount of academic research that is involved in the project.
I see. How fundamental is it?
STTR is more closely tied with academic research. So I don't know a lot about it. But anyway, so we do fund small businesses too, but with research grants, not with like equity kind of funding, which I don't know a lot about that kind of stuff. But my guess would be that that's not given to you to do a specific thing. That's given to you to make the company happen, right? We're giving you funding to do a specific project or create a specific product that is going to help you start your company or get something going or get a product to a stage where you can start to develop it into a commercial product. So yeah, we're a lot more early stage sort of investment.
And so the NIH, you can correct me if I'm wrong, I'm naive on this, they're in no way involved in trying to get things through like phase clinical trials. I mean, those are expensive.
Oh,
Do they get grants?
So we do fund clinical trials. My institute, I don't have a lot of experience with it. Like I said, I'm in the most basic research part of NHGRI. So, oh no. But the NIH does fund clinical trials. Other parts of the NIH for sure do.
Those clinical trials are super expensive, right? It's like phase 2 is 10 million or something.
They're expensive, and I like I said, I don't know a ton about it, but the other thing that I know about it is that one thing that makes it hard for clinical trials to be funded with NIH money, especially in a time like right now, where the stability of funding has been disrupted, right? For a clinical trial, you need to follow those people for a long time, and you need to have funding to do that. And right now, right now, sort of that stability of funding is one of the things that I think
Just want to point out you first tried that. That was
thanks.
perfect. Well done.
The stability of funding is very challenging right now. And even in like a normal sort of funding climate, it can be challenging to maintain that sustained funding for clinical trials or any other sort of long-term study. Because you have to we fund grants every 5 years, right?
Yeah.
And so you have to write a new grant every 5 years. And if you don't get the funding, then you have lost your ability to maintain what you were doing. But that's not super my area, so.
Yeah, no, that's fair. All right, cool, Rex. For your job, what type of emerging companies or trials in the genetic mutation market space do you think people are trying to solve and target the most for right now? Genetic mutation market space.
just gonna be thinking for a second because I mean, I can imagine. This is again. I'm not involved in the small business grants as much. Okay. Now this is not. I'm not going to do this the first time. In fact, I'm going to do this in many separate times. Some of the things that I'm sure that people are working on is like new detection methods or new like genetic testing platforms. Like, okay, well, sorry.
No worries.
Some of the way that we do genetic testing is through things like that are called like panel testing.
Oh, it starts you way back here.
Yeah. So you're gonna get to watch me do this all again. And so like panel testing is where they have sort of an idea of roughly the kind of genes that might cause the disease that you have. This is like when you have something more common, like cancer or something. We have a lot of genes that we know contribute to it. And so, okay, fine. And so I can imagine that some of it is like developing new panels for panel testing. Some of it is developing kits that allow researchers to do new kinds of assays in their lab. You know, you probably use kits, right?
Yeah, yep.
Yeah, so like a researcher will publish a new technique or a new technology that they've developed. Now, what's over there? And they want other people to use it. Oh, I have no idea what this is, and I can tell you I'm not going to achieve it. Probably have to get all the red coins, and that's not gonna happen. And so a lot of our, I know we have a lot of researchers in the small business portfolio that are developing kits so that you can buy that kit and then you can do that assay in your lab. That's not just for clinical use, that's also for research use. We also probably fund people who are developing sort of analysis platforms for okay, sorry.
Nice. Done.
Yeah, done that. There's probably gonna be another one here. Hey. Oh, okay.
Perfect. It's so stressful. You're relaxing. We're playing video games.
I know. I love it.
God.
I was looking back through to see who had done this recently, and I saw somebody was playing Metroid Dread.
Oh yeah.
That game stressed me out.
Rightfully
way
so.
too much. I gave up.
God.
I gave up.
Oh, So I played. There's only a few games my wife plays, and one is, I don't know if you've heard of It Takes Two. It's like a 2-player cooperative problem solving.
No. What do you do in it?
It's like well, the storyline is about divorce, but that aside, basically every level, each of you have an independent mechanic. Like one shoots the goo and the other one lights it on fire, and like you problem solve. And so it's not necessarily, there's a little bit of execution, but there's also, you know, just puzzles basically. And it is an outstanding game, but the sequel to it's called Split Fiction, and it is outstanding. If you're looking for a game for 2 players,
Is it on Switch? Or?
Switch 2 is
Okay.
Split Fiction, so you can get it for the Switch 2, and it is such an excellent game. But my wife doesn't have the like gaming prowess, so she just like hands get sweaty and she yells and panics every time something goes wrong.
I mean, I don't think that's
fun.
a failing.
No,
I think that is normal.
we beat the game. I mean, the game is beatable as long as you commit to it and execute. But now I'm playing with my 9-year-old daughter, and it's a lot of fun.
I've never played that, but we have a game called Octodad.
Oh, I've never heard of that.
It's okay. So that game is a dad, like a suburban dad, who is actually an octopus and has fooled everyone, I guess. But he's always at risk of being found out that he's an octopus. And so you control his legs and his arms separately. And that is also a cooperative game.
Each of you controls one of them.
Yes.
Oh my god. That's a special kind of hell.
Nice. Why did I do that? I think that you can. It's been a while since I played it, so I might be wrong. But I have like a memory that you can decide like whether one person does the arms and one person does the legs, or you might also be able to do it like one person does the left side and one person does the right side. I don't remember exactly. That game stressed me out a little bit, but
you played this with your kids or your significant other?
My significant other does not play video games. He does not.
Sometimes that's just divorce fuel right there.
Yeah, no, it's me and my daughter. The other one that we like to play is Overcooked. Have you played that?
I have heard of Overcooked.
that one basically ends up with us screaming at each other.
That's awesome.
I need meat. I need chicken. Some chicken.
Oh man.
Oh.
So have you been to the VR space in Railroad Square? The virtual whatever?
No.
It's like what used to be the Cap City Lounge and Proof down there in that area. But it's really fun, and my kids did it for a birthday, and I did it for the first time because I was worried about getting motion sickness and whatnot.
Yeah.
But their setup is really nice. I don't get motion sickness, and then my wife tried it and she loved it. So that's now our first Friday. We go there in VR as a family.
Oh no, I've actually only done VR one time. You've had Dawn Carr on this show before.
In fact, she's on in next week actually.
Oh, okay.
So she's back.
So her, this was like years ago, but her husband had a VR setup, and I don't even remember what we played, but we did play something at their house. And the only thing I remember about it was that I was so hot. I was so unbelievably hot, and I don't know why.
Yeah.
But yeah.
I mean, when it's that submersive, you feel it. That's awesome. All right. We have Double Damage. Beard looks good. Thank you, Double. Thank you for joining us. Welcome back to the stream. Yeah, the beard is something that was an accident. I got lazy at some point, and then my wife Debbie said she liked it, and so she's the one that has to look at me. So it's the compromise. Just let it grow out until I'm too annoyed, and then I shave it down and go back. But thank you, Double. Thank you for joining us. All right, QT Nat, do you have a favorite, most interesting epigenetic modification? Ooh. Wow.
I mean,
that's a very specific. My
CpG methylation.
My generic question is, do you have a favorite equation? This is a very specific version of that type of question.
No,
why is that your favorite, most interesting epigenetic modification?
Well, it's really just the OG.
Preference. Oh, okay.
There are so many different ones. So, one other program. Okay. I don't know how to get over there. Oh, wait, that thing is ringing at me. Okay.
Oh, nice.
I was like, I guess I'm supposed to do something. So one of the other programs that we are involved with at my institute is a new program that's run out of the NIH Common Fund, which I'll explain what the Common Fund is after I tell you what the program is. But the program is called RNomics, and it's actually developing ways to look at modifications of RNA, epigenetic modifications of RNA. So RNA, like DNA, gets modified in all kinds of different ways. We know very little about the scope of it because we don't have a lot of tools and technologies that even allow us to identify. So that's what I'm supposed to do. Okay, great.
You gotta aim them too.
Yeah, I can, as the cat, I can
Swipe,
spin my tail and... Okay. All right, sir, throw another one at me. Yeah, so we don't know a lot about it because we don't even have the ability to look at it in great detail. And we do know of some cases where changes in RNA modifications or errors in RNA modifications can be associated with disease. And there's probably more, and we would know that if we were able to even identify them and figure out what they do and how prevalent they are and in what tissues they're found. So the RNomics program is going to fund research that will develop new technologies that will help you identify them, figure out where they are, when they're found, and then allow you to do things like look at people who have disease and see whether there are changes in their RNA modification that might be associated with the disease. So that's a different kind of epigenetic modification. But yeah, there's all kinds of different epigenetic modifications.
Cutie Nuts follow-up. I love RNA modifications. So you're not alone. That's awesome. Kindred spirits. This is a fun battle. At least
yeah,
it's fun to
it feels like it's going on forever. Like, what do you want from me? And I'm small now, and you're throwing a lot of... Like, how long does this go on for?
No mushroom power-up.
It just, like, moves you to another...
We should do another prediction. Which one do we want? Oh, actually, Cool Rex has a follow-up before we dive... Oh, congratulations. There it is. GG.
Thanks.
Bowser's. Bowser smashing some alcohols. How we celebrate beating Bowser.
He looks pretty sad.
All right, Cool Rex has a follow-up. That's funny you mentioned assay and testing kits. One of my clients is a diagnostic multiplex testing platform detecting pathogens in blood. Very neat.
Right. Cool. That's awesome.
Yeah.
I'm curious to know what this person does with these people.
Well, Cool
like
Rex is a student at FSU. I don't. That was the original question. I think that's you, Cool Rex, right? A student at FSU, maybe grad student or med student. Who knows? Multiplex testing. So I mean, we're talking lab on a chip type.
Well, I mean, it's just really any way where you're creating a method that you can test multiple things in one experiment.
Do it realistically.
So for genomics, it's like they add barcodes to it, and then you can just sequence a whole bunch of different things all at once, and you can sort of differentiate it later in the analysis part by looking for the barcodes. So, anyway, multiplex can mean a whole bunch of different things, but it's basically just doing a whole bunch of assays in one.
Testing a lot.
Yeah.
Is that related? What was the company that overpromised and underdelivered and she went to jail? Theranos?
Theranos?
Yeah.
Did you watch that movie?
I did not watch the movie.
Pretty interesting.
Was the movie good?
Yeah.
It's worth the watch?
Yeah, I liked it. Or was it a movie or was it a series? No,
I think it was a movie.
It
was
was a
Amanda
Seyfried.
Yeah. I didn't, but like watching interviews with the actual, I don't remember her name. But
Elizabeth
like,
Holmes.
Yeah.
I am
the deep
pressing
voice, creepiness.
Oh yeah, she was real weird. But yeah.
Steve Jobs wannabe. Man, so that's worth the watch. I should.
Yeah, I liked that.
Cool Rex says, yes, I'm a student asking these questions because I'm working in a healthcare coverage group at a bank.
Oh.
Ooh, that is an interesting lens to look at healthcare.
Okay.
And it's unique to the United States, so. Oh, man. We should do a prediction. What one do you want to do next?
Well, I love talking about this.
All right, this one's presumably unrelated to your job. Actually, maybe. I don't want to spoil anything. But this is it's a fun one. All right. All right, ladies and gentlemen, if you're not followed, click the follow button. You will get standard internet units, and you can use those internet units to bet on a question that we are going to ask you guys. So I'm going to pop up a question. You're going to click the predict button, and you're going to put points behind which answer you feel is correct. These questions are written by our guests for the stream. And so the question we're popping up right now, cats can only make 2 fur colors, true or false? And so we know it's at least 1. We know it's at least 2. Can they only make 2 fur colors, true or false? I am genuinely curious where this question came from. And
Think of all the cats that you know and what fur colors they have.
True or false. So yeah, click that predict button. Use your standard internet units to gamble them accordingly, depending on your degree of confidence in the answer. But yeah, honor policy, just answer based on what you know. Oof, that was stressful. So what's funny is I never played 3D World, but I play Mario Maker, if you're familiar with that one, where people can make levels.
No, but that must get diabolical.
Yeah, no, it is. There are some levels that are impossible to beat unless you know a special code, but I love the technically hard ones. But that's the only place I've played as Cat Mario, like finding walls and all sorts of stuff. You're okay. Nice.
Yeah.
The bee bobbles. I like it. All right, you guys have about a minute to answer this question. Cats can only make 2 fur colors, true or false? I've never seen these characters before, though. That's fun.
Well, this is combining really having to be good at walking on things. Oh, with like
The 3D space.
yeah.
Yeah. Does the camera turn if you use the other joystick?
Oh, I forgot that I could do that.
Okay.
Yes, great.
Well,
Thanks for reminding me.
some of them don't let you do that. I don't know if this was one of them.
No, it does.
All right, cats can only make 2 fur colors, true or false? Lisa, what is the answer?
It's true.
Which is crazy.
Yeah, because think of all the cats that you know.
Brown cats.
Right. You have one cat?
We have 2. We have a black one and we have a calico one.
So the 2 pigment colors that cats can make are black and orange. And every other cat fur color is some variant of black and orange. And there are different genes that control that. So I'm just thinking of some of the cats that I've owned. I have had a Siamese cat that was a gray kind of point Siamese. A Siamese cat is basically a black cat. This one was a pretty pale gray, and so that is caused by another gene, which is actually you could look up what the actual genes are. I don't know those off the top of my head, but it's called dilute. So it basically, I think for that one, it affects the amount of the black pigment that's actually loaded into the hair cell during the hair growth cycle. And so less pigment is loaded in, and so the color is lighter, so it's gray. And then the Siamese, the color point where they only have sort of the dark fur on their ears and their tail and their feet, that is another mutation that causes the pigment protein to be temperature sensitive. So it's only stable, it's more stable in the colder areas of the body. So the ears, the extremities, the tail. And so that's why they have those color points. Okay, we don't need me's, but thank you. Some other cats that I've had. No, I don't. Because I don't even know what that means. So let's not do that.
Don't do it.
So you said you had a calico cat?
Yeah.
Okay, so a calico cat is black and orange. Is yours black and orange, or is yours gray and orange?
Gray and orange.
Okay, so the black and orange are those 2 pigments. Those are the protein is encoded by a gene that's on the X chromosome. It's not actually the same gene. It's different gene is orange protein and one is black protein. But the reason that they're patchy like that is because of X inactivation, which is what I did my PhD on.
Oh,
so female cats have 2 X chromosomes, male cats have 1 X chromosome. Female humans also have 2 X chromosomes, and male humans have 1 X chromosome. And in every one of the cells in a female, one of those X chromosomes is turned off. All the genes on that chromosome are turned off. And so it happens early in development. And then after that decision is made, it's transmitted epigenetically to all of the cells as those cells divide. And so you have like a small ball of cells that make a decision to make either orange pigment or black pigment. And then those cells divide and become a cat, and then that becomes patchy. And that's how you end up with a calico cat. Calicos are generally only female cats because you have to have the two X chromosomes for that to happen. Although there can be male cats that are calico, and that's because being male is actually dictated by whether you have a Y chromosome or not. And so there can be cats, just like there can be humans, that have two X's and a Y. In humans, that's called, I think that's Klinefelter syndrome.
Yeah.
But that's how you can get male calico cats.
That is intriguing.
Tabby cats, oh, and yours is gray because they also have that dilute mutation. And that makes the whole thing like just more muted.
If anyone's curious, I looked up the molecules that make this orange and black. There they are. The one on the left, this guy, is the black coloration, and the one on the right is the orange coloration. I'm a color chemist, so this is...
Okay.
I'm surprised I didn't know this, and I'm ecstatic that I do now.
Oh.
So it's just different amounts of these colors, basically.
Getting loaded into the hair follicle as it develops, right?
That's amazing.
What are some of the other ones? So like tabby, the striping. I'm not going to be able to explain exactly how that turns into like the tabby coat that you see, but that is changing the time in the hair growth cycle where the pigment is loaded into the fur. And so if you look at a lot of tabby cat, like actual fur, you'll see that there's like, oh, sorry. There's like a bands of different color on the fur. Like the whole fur doesn't grow in one color. It'll be one color down at the bottom, and then another color will sort of get loaded in, and then another color.
Yeah.
Anyway,
These waves of...
I know a lot less about how tabby works, but that is also like a coat color mutation. What are some other? Oh, white. White is not like white pigment. White is the absence of pigment.
Yep.
And
see right here, yeah.
Yeah, right.
I don't have gray hair. I have absence of color.
Right,
It's
exactly.
white.
So does your calico cat have white?
Yes.
So there's another gene that is white spotting.
That's fun.
And that controls the amount of white that the cat has.
And so the question for you is, why do we know so much about cat colors? Is it just the breeding? I mean, we're essentially controlling Punnett squares on a...
Because cats are cute.
That's
No.
number one.
I'm sure that a lot of it is because of breeding for specific things.
Yeah.
I mean, mice, so the mice that we use in research, they came from mouse fanciers who were basically breeding cute mice for pets.
Yeah.
And then understanding why I can't do anything else here. What do I have to get? Yeah, and so there's all other kinds of different coat colors that you see in mice and different kinds of phenotypes, and that's where they all started as mouse fanciers.
That's fun.
What do you want? How do I talk to you?
All right.
What is happening?
Cool Rex has an important follow-up. What do you think FSU will finish their football season this year? I'm going to be a senior now, and I can't bear to see us lose another season poorly. Do you have strong opinions about FSU football?
I don't know if any of my weather friends are watching this tonight. They were just talking about potentially watching it.
Yeah.
They all know I do not do sports ball. I don't know anything about it.
Sport.
I know it exists.
Based on your lack of knowledge, how will FSU finish out of, I think they have 12 games in the season?
Are we talking about football?
Yes.
Okay. Medium. They're gonna finish medium.
So 6 and 6.
Sure.
There you go, cool Rex. Man, those are my favorite bets, actually. I know. So I played football in high school, and it was hard for me to football because I got hurt a lot and I got injured a whole bunch, and I despised it for years. And then I arrive at FSU in 2013, and they win the national championship. And it's like you can't get away from it here. It's just everywhere.
Yeah, that's one thing that I find very, let's say, interesting about living in a big college town.
Yeah.
I went to Case Western Reserve University, the famous engineering college.
Yeah. Not known for their sports.
Not known for their sports. We did have sports, but not a big thing. And so having so much focus on sports is very new to me, and I find it very fascinating. I find it very fascinating that like stores sell like going out clothes in school colors. Like the whole thing is not my college experience.
That's fair. So your kids are not into sports then?
No, they're not. Although my husband, now how do I get there? My husband is English, and he and my daughter just went to England. They were there for the World Cup.
I was going to say that.
The end of the World Cup.
Yeah. Speaker.
And they watched I think they watched the last 2 England games there. And my daughter got very into soccer at that point.
And that last game was fun, though. Did you watch it? The bronze medal World Cup game?
I think I managed to watch the end of it.
It was like 6-4 or something ridiculous.
Because of my non-sports ball watchingness.
Yeah.
I don't subscribe to the TV that allows you to watch sports.
Yeah.
So I had to go to deep. and I can't remember if this is the game that I saw like very little of it because I didn't make the decision to like leave the office and go to deep until the game was almost over.
Yeah.
And then by the time I got there, actually when I left, like England was winning by multiple points.
Yep.
And then when I got there, England was losing. And I was like, I swear it was only 15 minutes that it took me to get here.
That's fun.
Yeah.
It's like a watch party, though.
Yeah. So.
I like it. So, Cool Rex, to answer your question, we don't have an answer to your question. We'll see. I've become quite jaded with college sports in general because of the dollars and the transferring schools. And it's just, it's lost its, I don't know, the purity associated with it, maybe? I don't know.
Yeah, it seems like from an outsider's perspective, it seems like a business.
Yeah, no, it's professional football light.
Yeah.
It's definitely changed allegiances and who you can like buy your way to success, much like professional teams. But Lori said Case Western, Ohio represent. Shout out to Case Western.
I went there for undergrad and grad school.
Oh, that's awesome.
Okay, I'm at a point in this game where to do better, I'd have to pay better attention and find 2 more stars, it seems.
No worries.
So maybe I will change it and try the surgery game.
Let's do it.
Okay.
Let's do it. That one's pretty mindless, right?
No, yeah. I'm only doing surgery.
Lori's question is, are you from the Midwest?
I am from the Midwest.
Where are you from originally?
I am from Racine, Wisconsin.
Wow, I'm from roughly St. Cloud, Minnesota.
Oh, I think I remember that.
Midwesterners.
So if you've ever had the wait, I just have to figure out how to get out of this game.
I think it's closed software, right? Left.
Oh, yeah, okay. If you've ever had Kringle at Trader Joe's, have you ever gotten the Kringle?
Yeah.
That's from Racine. It's from O&H Bakery in Racine. So I've eaten at the bakery.
That's-
Oh, here are the me's of my
Oh,
family.
you want to show them? Sorry, I didn't
Well,
say
I made them a long time ago when my daughter was a child, and so she's got pigtails and stuff.
Oh man, how does she feel about? She hasn't seen it.
She has seen it.
That's awesome.
She thought about making a new me, but she liked it.
Racine women's baseball,
Yeah, that's right.
Yeah.
It's in that
league of their own.
yeah.
Yeah, that's fun.
But anyway, that's where I'm from.
Oh, and Cool Rex has redeemed Take a Drink, so Lisa, cheers.
Okay.
Thank you for joining me. Cool Rex, hopefully you have a drink in hand. You don't start class until Monday, so you might as well relax right now.
Now, do you really want me drinking wine before I'm about to operate on people?
Oh, let's do it. They're soulless people. Do you want more or you're good?
I ask you. Sure. Okay. Point the Wii remote at the TV to use this.
All right, so we've done a surgeon simulator before. Surgeon simulator, I don't remember what
really?
it's called, but it's a mouse-controlled game. And Molly Hermiller, the neurobiologist, showed us the parts of the brain via the game.
Oh,
It's
really?
kind of fun. She got to do brain surgery. Cool Rex has redeemed Take a Drink again, so cheers.
Okay.
Another sip before we do some surgery.
Select try again on the TV. All right. I'll do that.
Yeah, we got it earlier.
We did. Yeah, we tested this to make sure it was going
I'm
to work.
confident. You have to turn off that screen or something, or go home on that one. It's technology, man.
Yeah, I really, the Switch is so much easier.
Yeah, exactly. It just communicates. With that said, this was cutting edge. This walked so Switch could run.
Oh, it's true.
So, shout out to the.
I mean, just the like ability to have this like.
Yeah, if anyone hasn't, you should show them that on camera. You can see right here what they
Oh.
can see.
Yeah.
Just show them what that like controller
yeah.
looks like.
It's like
Yeah.
the most primitive handheld. What I don't like it. It's not very comfortable to hold.
Yeah, it's not
Point
ergonomic.
The Wii remote at the TV. Use TV only.
Hmm?
Um.
Do you have to turn that guy off?
No.
Should be on. Schmo Knight, that looks clunky. It is clunky.
It is pretty clunky.
Compared to the Switch, for sure. Switch is pretty sleek and feels much
Okay,
sturdier. Than
here's
this
what
thing.
I'm gonna do. I'm gonna do the time-tested
Punch it.
solution of turning it off and turning it back on again.
Just blow on it.
So we're gonna just start over.
I like it. But yeah, if you guys have any questions for Lisa, we're about an hour and a half into the stream. We've journeyed through a couple different Mario games. Now we're going to do a little bit of surgery, assuming the Wii U behaves. But if you have questions for Lisa, she's happy to answer them about. We're talking about genetics, epigenetics, genomics, whatever it might be. She's also a deputy director at the NIH, the Genome. Sorry, I don't remember the name of it.
National Human Genome Research Institute.
There we go. So if you guys have questions about that or the NIH or whatever it might be, we are happy to talk about it while we troubleshoot and deal with a little bit of WiiU trying to do surgery. We got it to work. Come on.
We did. Why is it? I don't know what you want from me.
Does anyone know what this means? The Wii U menu could not be opened. Select, try on the TV again.
Well, hope you're enjoying this riveting gameplay.
No worries. But I should ask you a few questions. So we're
Okay.
so we talked about the Theranos movie.
Yes.
I think I asked you this last time. I don't remember what your answer was, but it's one
Are
of
you
my
gonna
favorites.
ask me?
Movies or TV shows
Oh.
that get your discipline right or wrong, or get
Oh.
genetics right or wrong, or any variation on that.
So, I actually I don't watch a lot of TV shows anymore
Mhm.
that where genetics comes up. but I have in my lifetime watched like, this is very frustrating and I don't know how to solve this problem.
All right, let me just Google that next time that comes up.
We tried this before.
No, it
we
worked.
started.
I saw
Make
you
sure
do
that
surgery.
it worked. You did.
You got so many cools.
I did.
Oh,
Oh, okay, great.
we're good.
I think so. Great.
Oh, that's exciting.
Oh yeah.
Ask a Scientist tech support. Absolutely, Schmonite. We're ragingly incompetent here. We need help. Have you tried turning it off and on again?
I did. That's the first thing I tried. It's my go-to move.
All right. Do you have your safety strap on?
No.
Oh man, I love those early videos when this came out and people threw controllers at screens and smashed them.
No, yeah,
Not that aggressive of gameplay. But yeah, you should show them the nunchuck too if they're not familiar with this.
Yeah, you out there probably have never seen this. This is actually one of my favorite game controllers.
Yeah.
Of all game controllers. It's so comfortable.
Yeah. And one of them has the D-pad and the other one has this joystick and triggers on both.
No,
like you can do shooters with it. Yeah.
I love it. Okay.
All right. Here we go.
All right. So this game is it's basically like a soap opera type sort of game of some sort. I'll probably...
Oof, that's probably pretty loud for you guys. I apologize if you guys got washed out. No, that's not your fault. That's just different systems have different inputs.
I think because I started playing this again with my daughter, I think we'll be able to skip past some of the TV drama aspects of this.
Oh, is this the game where you stitch people up as you warn on your relationships?
Well, I'm gonna do some amazing medical care here, all kinds of things.
Oh man, Lori just said, did you just cousin Vinny us with the Utes? That's amazing. Also, the appropriate age for us to get that reference. Utes? Did you just say Utes? Youths.
Okay.
Youths.
The Utes.
The Utes. The two Utes.
Time is of the essence. Let's get started.
Yeah.
Oh my god. Anime drama and surgery?
Yeah.
This is amazing.
Yeah, so I haven't gotten far enough into this game, like, again, to remember sort of what's happening. But this doctor has some special power that sometimes comes into play. Yeah. Okay. Oh no. This is the nunchuck that doesn't work. Let me go get the other one.
Oh no. Vitals are fading. Run. Oh no. Heartbeat dropping. It's fine. Patient care. Those sessions did nothing for you.
You win some, you lose some.
Yeah, that's fair.
You do need the nunchuck to work because that's how you control what you're doing.
So the I don't remember what it's called. The wand controls where you're positioned on the screen.
Yes.
And then the joystick.
And then the joystick controls. See that like flower over there? So each direction of that is like a different tool or thing. So
I see.
Okay, so I want you all to watch how badly you can sew and get cool is the best score.
As long as you get
I
across
just
it.
got the best score for that stitching. I do not have a career as a surgeon.
Schmonite said, we used to draw silly pictures with the stitches.
Oh, yeah, that's...
Still get a cool.
Yeah.
Speaking of Cool Rex, thank you for joining us. Cool Rex says, thank you for the chat, guys. Have a great rest of your evening, and let's have a great first week back next week.
Okay.
Man, so that's annoying for you. Like when students come back, parking becomes a nightmare for you as
so this is gonna be.
Oh no, you have a spot?
Whoa, yeah. Actually, the weather office has their own parking lot. it's like a small little lot
That's awesome.
that's behind another lot. So I don't have to worry about parking.
Other people don't know about it. Is that?
Well, you have to have access to it. Like it's
Oh, it's specific. It's not
hard
faculty
control.
document.
No,
It's. Oh, nice.
okay. I'm going to close up some of these. What you will learn in this game is that antibiotic gel can do everything.
I mean, it is a prevailing medical opinion right now, right?
Yeah.
Throw antibiotics at all of it. It'll be fine.
Yeah, so parking isn't going to be a problem, but I understand from everyone that my commute is going to change.
Oh, man. Well, especially with all that construction.
Now we need to reconnect
Yeah.
the bone fragment.
Be a
Twist your
nightmare.
wrist
Okay.
and maneuver the fragment into the correct position.
Okay.
You guys will have to let me know if the volume's too loud or if it's acceptable.
Now, this is...
That's not
Yeah.
the correct location.
No,
You
it
have
is
to
the
find the
correct
right spot.
location. Yeah, and you have to have your hands be steady enough.
The patient's vitals.
Yeah, the patient's vitals. Sorry. Let's give him some of this green juice that's going to get his vitals back up.
This game is great.
Okay, we're going to try this again. Now, I know where it goes. It's just keeping my hand steady enough.
You've successfully
That it will
replaced
go
the
there.
first
Wow.
bone fragment.
This
You're
one
supposed to figure
goes...
that out.
Yeah, oh yeah, it's really...
That's a little janky.
You really have to do that one first, too, because if you don't, there's like no way you're gonna find these other ones. Okay. And then you like hurt this person every time you get it wrong.
I mean, you are trying to jab bone in there. Oh, that's great. All right. Anyway, movies or TV shows, genetics.
Oh, so I would say that my memory is of watching shows like CSI or like I remember watching the X-Files
Yeah.
at some point, and they like, first of all, they're running these things in like a very futuristic looking lab where
That does
they're
it.
like
Let's close them up.
looking at like one of those TVs that's just on a glass wall.
Yeah.
And it looks all fancy and there's like neon light and everything. That's not what it's like. And then they say, I see they have a gene.
We need to bandage
Yes,
the sutured
as we've
incision.
discussed, everyone has a gene.
Use the antibiotic gel to
So
stop the
that's
bleeding
probably it. I
before
think also
bandaging
like the
up.
speed at which those things happen in the shows is not at all consistent with
The
reality.
bleeding stopped.
That's fair.
Hand me the bandage.
But
Apply
that's
the bandage
probably
from one end of the sutured incision to the other, and we'll be finished.
that's probably
Good work,
the example
everyone.
I would give. All right, I saved that guy.
I gotta. Wife needs a code for something. All right. Hopefully that took care of it. But yeah, CSI is amazing for all things science. Science loosely defined. Also, classics
Wait, I
like
just did that one.
Gattaca, genetic engineering.
Yeah, actually, I think I did see Gattaca, but I don't even remember much about it. So
Wow, Lori, this is a very specific one. Love CSI. I knew I was old when I realized I thought Gibbs was hotter than
I'd
Dinozo.
appreciate it if you could arrive on time, Marcus.
I don't know those people, but I'm
You
also
didn't know the
old, so I
details.
probably would
Yeah, I'll agree
have
on
the
that
same
front.
opinion as you.
Of course, Dr. Vaughn.
TV shows are a nightmare for STEM field people, absolutely.
So as a
I mean, but
precaution,
you
we're
gotta like enjoy those precious moments when they get it
Oh,
right.
Dr.
When somebody
Vaughn.
actually
Right.
did their
We're not letting
homework.
the
I do
patient's
think
rude
that
comment bother
It
you, are-
seems like
I
a
guess we
lot of
just have to
shows
show him how talented we
lately,
really are.
well, not maybe not lately,
Yeah,
but like
you're right, Val.
do try to get it right. Like they actually like consult with scientists. Okay,
Oh, and Lori said, "Never mind, I got my letters wrong." NCIS.
that's the military one, right?
Yeah,
Okay.
I'm assuming. I don't know the.
Okay.
I think
Let's
that's the one with like
begin the
Mark
operation.
Herman.
Disinfect the area with antibiotic
Okay,
gel. and
antibiotic
clean him
gel
with your
does
incision with a
everything.
scalpel.
Including starting the procedure. Okay. You don't have to cut them very nicely, and you still get a good score for that.
Small tumors cannot spurn them before we
Oh, we're gonna
extract the
burn
larger
some
ones.
small tumors.
Double damage.
A laser.
Get it right when? I mean, this dude goes into a black hole and travels back in time. I mean, there's some great ones, like, I mean, examples like Interstellar captures, like, the black hole and the math. They actually simulated it based on physics models and things like that. One of my favorites is Real Genius, this, like,
Oh, that was
late 80s.
Yeah,
But
I don't remember very much about it. So if
But
you
they-
can remember it, tell me about
they
it.
designed lasers. It captured college graduate life pretty well. But they talk about, you know, this energy, potential energy surfaces and coupling to states, and like they did their homework, they did research. I mean, other ones, Breaking Bad is an excellent TV show that captures synthetic chemistry pretty well. Man, one of my favorite ones recently is Godzilla -1. Have you seen this one? It's like a Godzilla origin story. It's one of the more recent Godzilla movies.
No, but I think I heard them talking about it on a podcast that I
Yeah,
listened to.
because there's a new one that's like Godzilla Zero, which is the sequel to Godzilla -1. I think those are the names. But Godzilla -1, the way they trap Godzilla, because you can't just nuke him or kill him. Spoilers, anyone that hasn't seen him. The idea is to put him on a platform and then create bubbles on the platform so he sinks to the bottom of the ocean. And the idea being he can swim in water, but if you have bubbles, you're breaking the surface tension so he can no longer swim. And so he's trapped at the bottom of the ocean based on this bubbling platform. And their backup plan, if Godzilla does somehow the bubbles screw up, raise him to the surface really quickly and give him the bends. And so don't give him time to deoxygenate and degas the blood. So it basically rips him apart from the inside. I'm like, that is some solid scientific, like, fundamental general chemistry. So, rant done. There are a few good ones out there. But yeah, genetics were... I read this portrayal of science in science fiction and art. And it's basically... You see this trend of whatever's hot and scary at the time becomes the pivot point for discussion.
Is it like CRISPR engineering?
Well, at some point it was, but like, so if you look back at like comic book characters, like first they went to the African jungle and got bit by something and they got superpowers, right?
Yes.
And then things like nuclear testing came in, and all of a sudden nuclear mutants were a big thing. And then genetic engineering took over, and now it's like, I don't know, AI or machine learning. Like we are a byproduct of our fears of the modern science.
That's interesting. I never thought about that.
And so like the X-Men fell in this particular range where human genome was being solved, sequences were known,
Right.
and that became a pivot point. And same thing with like Jurassic Park fell into that window
Right.
of genetic engineering. So there was a bunch. Whether they got it right or wrong is another debate, but became the topic.
What are your thoughts on the Big Bang Theory?
Oh, I hate it.
I hate it too.
It is nerd blackface, basically.
That's a great way of putting
Yeah,
it. I
that's-
call it a caricature of scientists.
Oh yeah.
Like a very insulting caricature of
No,
scientists.
absolutely. And I saw a really great critique on it that there's the phrase was adorkable misogyny, where the main character can be sexist as long as he's this weird awkward guy. He can like, you're pretty good at math for a girl, ha ha ha, and they get away with that in that show. And so yeah, there's many things.
Now I just want to point out, because I've been skipping all of the lore like parts of this, you've missed this like new nurse coming in here, but she's wearing like a lace camisole to surgery. I just wanted to point that out. Also, this hospital, you missed this part, it's in Alaska. This is her work wear in Alaska.
That's amazing.
So I just wanted everyone to
You guys need to know the backstory. But anyway, enough rants and detours. If you guys have questions for Lisa, she's happy to answer them. Let's talk about whatever you guys want to talk about.
Except the Big Bang Theory. because
Except
we
for the
both
Big
hate it.
Bang. I mean, we can rant about that all day. I've watched parts of it. I've seen clips, but like my favorite clips of it are when they remove the laugh track and it just shows how uncomfortable and unfunny it actually is. Because laugh tracks work. That's why they did them, right? They're social pressure to find jokes funny, right?
That's
It's
true.
easier to go to a show and find jocks funnier than if you're alone. So, God, I should stop ranting about it. Lori has redeemed a request to factoid. Do you have a factoid, a tidbit of information, a knowledge bomb you would like to drop on the audience? Can be
Like-
anything related. We've already talked about cats, so
I didn't think about that ahead of time.
and I didn't remind you because you're a returner. I mean, well, let's do some factoids about.
Synthetic wet membrane. Okay, sorry. Hang on. Let me just figure out how to cure this person. Oh, his vitals are so bad.
Now, let's treat the remaining wounds.
I'll treat the remaining wounds when I save this person and make their vitals like. I think I can't remember if I had this as one of my things, but like
Oh, you
what
can steal one of
percentage
those.
of the genome is actually coding for protein?
Yeah, let me go ahead and use it.
It's like 2%. Like most of the your genome is huge, and only a very small amount of
All
it
the
actually
external wounds
like
have been treated.
makes
Let's open him
the
up.
proteins. There's all kinds of sequence that is between those that we know that there are parts of that have functions. They're called regulatory elements. Okay. This is where it's going to start to get hard because this person is going to be dying, and I have to drain all of these blood things and then find. Okay, so let's.
You located the
I
gunshot
located
wound.
the gunshot wound. Thank God.
We're dealing with his heart, so please be extremely careful when you
Oh, this is his
extract the
heart?
round.
Oh no.
Oh. Okay, wait.
Let's drain the blood and determine the
Okay.
mistake.
Oh my gosh, he's gonna die.
Please extract the round carefully.
Oh no.
Oh, your skills are not up to the task.
Oh no.
What I love about this too is it's like, and these doctors gave up on being doctors. It haunted them forever. Yeah, I've ruined everyone's life now. The doctors, the patient.
That is amazing.
Yeah, so actually, I took my daughter up to Bethesda this past weekend, and we visited the NIH campus.
Oh wow.
And I took her to the NHGRI part of the NIH campus. And there's a wall that they've put like the sequence of a very small portion of chromosome 7. And the whole sequence is on this wall. First of all, it's huge.
That's awesome.
And then they've also like indicated what parts of that sequence are the protein coding regions of the genes in that region. So there's actually like another part of this that shows you the genes and the exon structure. And then it highlights that on the walls. What's striking is that there's so much sequence that is nothing.
Yeah. Or unknown.
Or unknown, right.
Yeah.
Treat the external wound.
Yeah, those of you not familiar, DNA writes, well, all sorts of things. It's an executable. And some of those things are proteins. Proteins are like the workhorse, the enzymes, the whatever it might be that run aspects of the body. But a whole lot of DNA doesn't do a lot as far as we know.
Yeah, we're learning. So NHGRI had a program that's called ENCODE.
Let's open him up.
That had a goal of identifying functional elements in the genome and sort of characterizing what is happening in those
He's in bad shape.
non-coding
Please keep an eye on
regions.
his vitals
So
while
there's
you
things
treat the
like
wounds.
promoters that, help genes get expressed. There's things like enhancers, which basically tell genes the time and place in which to be expressed. Okay. This is where things got...
You
Yeah,
look
no, it's
dealing with
pressure.
his heart, so please be extremely careful when
Yeah, I'll be
let's drain
extremely
the blood and
careful.
determine that a mistake could be fatal.
Now, don't let me let this
Nope,
man's
13.
vitals go down. Okay.
10.
Yeah. Oh, I've got to get the. That's right. There's a gun something in there, and I got to get that out. I need the forceps.
What?
What is
Split
that?
in two. The other half must still be live.
Oh
and we
no.
have no choice.
Oh no. We have
to
no
open-
choice. Okay.
Oh, 10.
We can't
That
get a
one low
pulse with
quick.
the round still. Please extract it quick. Open up the gunshot wound with the scalpel.
Okay.
Keep an eye on his vitals.
5, 4, 1. Done. Operation
It
failed.
appears I made it in time.
Further that time.
No, that was good. You got the thing out.
Yeah.
But then it dropped real quick. It went from like 30 to 10 for some reason.
Bringing them much Dr. Vaughn and Dr.
Yeah.
The stress and drama of this.
Never.
But if you guys want to make it more stressful, ask Lisa questions while she's trying to do surgery. We just crossed 10 o'clock. It's been 2 hours of stream. Time has flown while we're having fun.
Well, we're having fun.
Yep. So I gotta look up. Do you remember what your NARC time was last time?
Okay,
No.
let's give it our all. We need to treat the external wounds first.
I'm gonna look it up.
I am very competitive, though.
Yeah,
and
no, I respect that.
Fix it now.
So do you want to know what your previous time was? I think you should.
No, I don't. You can tell me after.
All right.
What I remember the most about the last time I did this was I played Metroid,
Yep.
which is like one of my favorite games of all time.
External wounds have been treated.
And
Let's open them up.
it was so hard for me to not just like really try to do good at Metroid.
It's the pride behind the game. Speaking of Metroid, I got fun news for you guys. So Lisa came bearing gifts. She got me a copy of Metroid Yellow. You guys can see the cartridge on the screen right there. This is awesome. I didn't have this cartridge previously. I have Metroid, but not this particular version of Metroid. And what's really fun about this is I now have 2 copies of Metroid for the Nintendo system. And you guys can see the cover art varies on both of them. And so the backstory about this, and at least the reason I was excited about this particular version is there was a downfall of early video gaming. I don't know if you heard, like, after Atari and ET was like, the market crashed basically entirely.
Right?
And it was one of the things that they had a problem with is you had cover art that looks something like this, right? ET cover art looks like that.
Yes.
And you go to the game and it's just like 4 squares moving around on screen. And so the art was very deceptive to what was actually sold in the game. And so Nintendo was very strategic in their early games. And they basically showed the gameplay directly on the cartridge. And so early Nintendo games did this. They were very strategic in showing what the game looked like. But then later, when they re-released, or they released Metroid 2 on the Game Boy, they really released the cartridge and went back to, you know, classic cover art, like the romance novel depiction of what the video game looks like. And so, yeah, 2 generations of Metroid cover art.
So I definitely had the original one
Yep.
when I was, you know, a child playing my like Super Nintendo.
Yep.
I think I got the other one. I bought another Nintendo system when I went to college. I had it in my dorm room.
This would have been the...
And I'm pretty sure that I got that then.
Yeah.
Yeah.
No, that timeline's right. So, yeah, there's the newer cover art and then the original cover art. So, yeah, I now own 2 versions of Metroid, and I'm super excited.
Okay,
All right.
this is
Last try.
probably getting too stressful.
It's up to you. If you want to do something else, we can. But yeah, you guys got a Nintendo cartridge cover art factoid free of charge by me because I nerd out about those things. All right.
Maybe we just skip that one.
Yeah. An aging hospital. Oh, is this one for Don Carr, our guest in 2 weeks?
Oh, no, I think this is like the actual hospital itself is old.
Oh, not the people.
Not the people in the hospital.
The CAT scan we took earlier.
You have to use
has
like
confirmed
caveman tools.
Right. It's
Just rocks and
it's
twigs.
this hospital in this remote place in
The
Alaska,
objective of this
and
operation is to extract
they
these
want to close it.
It's progressed this far.
I'm concerned about his physical condition. He's not.
I like it. All right. We
You're the
redeemed
only ones
Lori's
that
request of
don't worry.
factoid. Yeah, if anyone has questions, Lisa's happy to answer them. We can talk about the NIH. We can talk about life as a program officer and now deputy director. Talk about genetics, genomics, epigenetics, whatever it might be. Rare diseases, which is a fun the descriptor. An intriguing topic.
Yeah, I had so many ideas of things I could talk about related
then you
to
forget
that,
'em.
and then like when I'm playing these games, it's
Yeah. What's your,
Those are located
I don't know
in
if
small
favorite's the
intestine.
word, rare disease. What rare disease do you think is most interesting that you've stumbled across?
So I actually don't do much with the actual diseases, right? What we do at our institute is develop ways to find the mutation.
I see.
We actually don't focus on any specific disease. That's actually like, no, that's this one.
It's like a treatment.
That's
of
actually
things.
part of like the mission of our institute is that we develop technologies, tools, and methods that can be
Let's continue
applied
with the remainder of the
broadly to
operation.
any disease.
We found a
So
tumor.
in the
Let's
Gregor
use the
program,
power
we
system
don't
here.
focus on, specific diseases. We focus on figuring out ways to diagnose patients regardless of what disease they have.
I see.
Because they're more about like figuring out the method to do it than the actual disease itself.
The tumor has been removed.
Oh,
Please
that makes
paint the
sense.
area where you may be
Now,
excited.
taking the tumors out, I can do that. That's really easy.
What is the name of this game?
Trauma Center. I think this one is New Blood.
Trauma
There's
Center,
more
New
than
Blood.
one. I have another one too, and I tested it out before I came here, and it doesn't work as well.
It looks like
I think
that's
it's
everything,
older.
but his
Trauma
vitals aren't
Center,
stabilized.
Second Opinion.
I have second opinion, yeah.
And Trauma Team is another
None
one.
of the
Oh,
That's
preliminary
awesome.
scan missed anything.
All right, I got to change our title because people really want to know what's being played here. So Trauma Center, New Blood.
Yeah.
There it is.
Let's pump up these vitals before. Oh, wait. No, we probably just need to. The magnification tool. Right. How do I do that? I forgot. Oh yeah,
This one
that's
over
right.
here? It's progressed much further
Okay.
than we expected.
I
We need
like
to
it. So related to testing and assessment, or could be related. Unlimited budget, no moral qualms.
I don't like answering that question
Why?
because moral qualms are very important in human research and genetics research. You asked me that last time,
Yeah,
and
I
I
did.
dodged it last time too for the same reason because I don't.
I mean, Sarah Hart didn't dodge it.
Yeah, I'm not Sarah Hart.
she went all out
no.
with making like octuplets and separating them.
Oh,
Now
Yeah.
treat the-
So you could do it in a reasonably moral way, but I get it.
no,
I completely
I don't like.
understand.
I don't like.
Well, let's leave it at unlimited budget. Moral qualms are up to you.
I mean, we're-
Contacted the
so-
University Hospital.
Okay, I don't think I can murder people anymore.
All right, so what do you want to do?
Well, I did mention to you that we could play Dr. Mario.
We could play Dr. Mario. Should we play Dr.
I think
Mario?
we should play Dr. Mario.
All right, let's do it.
That's what I would say.
Have you played Dr. Mario since then? 'Cause
No.
I have not either.
Okay,
So we're starting.
Yeah.
So anyone that's not familiar, Lisa and I are arch rivals in Dr. Mario.
Right.
In fact, of the 100, this is the 131st stream, and probably the 80th something guest. And I've only played with a guest once before, and it was Lisa because she challenged me to Dr. Mario.
Yeah, I mean,
And so we have to revisit the rivalry right now.
now, okay, can I play by myself first?
Yes.
So that I
go-
can
go-
remember how to play.
that is completely fair.
Okay, thanks.
Those of you not familiar, Dr. Mario, why are you not popping up on the screen? NES.
It's here, but it's also not controlling it.
Interesting. Let me try that again. I might have used the wrong emulator. All right, now it should work.
Okay, I just need to remind myself how to play it.
All right, anyone not familiar, Tetris is a way more well-known game, but Dr. Mario is in that similar vein. The idea is you have viruses on the screen. You have to line up 3 color portions of a pill on top of those viruses to kill them. You need to line up 4 of a color. And
Right.
then when you
Okay.
do that,
Okay.
you ready for this?
We're already like seeing that this is gonna go badly
Oh,
because
you
I'm
in like
trying
medium
to.
speed
Okay.
too.
Yeah.
You
Yeah.
just go
Okay.
slow. Just go. Yeah.
You're right. Let me ease into this a little bit more
PTSD.
while I
from your
remember
Ask a
how to
Science
even
questions.
use this controller.
Oh man, it's a crippler.
Okay.
Well, yeah, I have low speed. Yeah. Music though,
Oh yeah, I
The
set
music
the
are
music
bangers
to
on this.
chill.
because we need to be chill. This is stressful.
Yeah. Okay. This is. This was a
I
much
mean, I think
better
what was
idea.
unfortunate for you last time, it was coincidentally before you were on the stream, is when I got like the speedrun times on Dr. Mario. Like I had this like 6-month kick where I went back to all my childhood games and decided to speedrun them.
Yeah, you really killed me, but it was fun.
Yeah.
I used to play it a lot.
Oh man, I love this game. This is one of my all-time favorites.
no one watching this stream would look at me playing these games and think I was good at any of them. But like, I actually did used to be.
I mean, I think the hard one about Dr. Mario is if you only played single player, you didn't have to deal with the drops from the other player.
Right.
And that becomes a pain in the ass real quick.
I remember very vividly being reminded that that happened.
Yep.
Because I was playing with you.
Yep. And like when because my wife and I used to play this all the time, and we would strategically shit on each other all the time with kills.
Yeah. Well.
But yeah, as Lisa's trying to focus, if you guys have questions, she's going to be happy to answer them.
I'm gonna try. So I was gonna talk about one of the programs that we support at NHGRI is we're really big on acronyms at the NIH.
I mean,
And so this one is called IGVF. That stands for Impact of Genomic Variation on Function. And what that is trying to do is really look in a systematic way at making mutations all across the genome in a like very densely, right? And figuring out what those mutations do to the function of the gene and the cell and whatever, right? One reason that we started a program like that is kind of about rare disease, right? So one of the reasons that it is difficult to diagnose people with rare diseases is that you find mutations that are only found in the person with the disease and not found in normal individuals. And you think that those could be involved, but you need some sort of supporting evidence to convince you that that's it. And so a lot of times when you get genetic testing, what you'll end up is you'll just get, well, we found a mutation here, but we don't know if it's significant. They call it a variant of unknown significance. And you need like some sort of additional evidence to classify that more concretely into being either a pathogenic variant or a benign variant. And so that's one thing that this program is doing, is generating some sort of functional evidence about the effects of different mutations or genetic variants and how they impact the function of the cell. So they're mostly looking at sort of molecular profiling. So they, but also other kinds of like phenotyping. I would say a lot of it's molecular phenotyping though. And so one example where something like that is useful is, this was in the news. Actually, I don't remember exactly when this was making the rounds. But Chris Evert, who is a tennis player, that's a kind of sports ball. She's a famous one.
Yeah.
She has ovarian cancer, and her sister had ovarian cancer as well, and died from it actually.
Oh.
And because of her sister's cancer, she was encouraged to get genetic testing. Maybe her sister had the genetic testing first. And they had found in the BRCA1 gene, which is known to be associated with ovarian cancer and breast cancer, potentially disease-causing mutations, but they couldn't classify them definitively as being pathogenic or benign. And so she just had variants of unknown significance in the BRCA1 gene. And then what happens is they'll go back over time and re-look at variants of unknown significance and see if in the meantime additional data has been generated or published.
Oh, interesting.
That can help you classify it. And so I think what happened was then she like found out that they reclassified one of these VUS and determined that it was pathogenic. And so then Chris Evert was able to get the genetic testing, and she found out that she had that variant. And so she actually caught her ovarian cancer much earlier than you typically catch it. Because I think I don't know a lot about ovarian cancer, but I think it's one of the kinds of cancer that you don't often know that you have it until you are really far along in its progression. One of the groups that is in the IGVF program is doing what are called, MAVE assays. It's basically a very saturated mutagenesis of a specific gene that's known to be associated with a disease. So they've looked at BRCA1, for example. They create mutations like all across the entire protein and as many combinations of mutations as they can.
Jeez.
And then they do different kinds of like molecular phenotyping or different functional assays, in an experiment, like not in a, like in vitro.
Yeah.
And they then can use that to try to classify those mutations as having phenotypes that are consistent with what a benign mutation has or a pathogenic one. And then when you find new mutations, you can look at how they perform in those assays. And then that gives you some additional information about whether they might be benign or pathogenic. That's really hard to explain why.
Yeah, while Dr. Marioing.
But I
Don't
hope
worry,
that
you're a
that
doctor.
made sense.
You're a doctor. It's fine.
Hope that made sense.
I like it. But so, I mean, genetic sequences, you're intrinsically dealing with a whole lot of information.
Yeah.
And something that has ramped up dramatically since the last time you were on the stream is this machine learning AI. I mean, how has that invaded your space in terms of a program officer, but also as, just in general knowledge in the field?
Yeah, I mean, that's definitely where things are going, right? Now, I am very famously not a computational person. And so I don't actually get grants that are using those approaches or developing approaches like that in my portfolio too much. So I'm going to sound like a real noob when I'm talking about this.
Yeah.
But there are definitely a lot of computational tools in all areas of genomics that are using machine learning and AI to help crunch data, to help interpret data. We just released some funding announcements for the next phase of our rare disease program. One of the things that we are looking for is people to develop new computational methods that can help people analyze whole genome sequencing data to try to identify variants that might cause the disease. And we expect that there will be, applications that will come in that will use AI and ML.
I mean, so along that vein, and harkening back to the original, unlimited budget, if you had basically the genetic sequence of every American plus the disease record of them,
Yep.
is that enough to solve for a lot of these rare diseases in terms of pinpointing genetic correlation with outcomes? I mean,
That does give you a lot of information. I'm going to try to talk about this intelligently while I'm doing this.
yeah, no worries.
So, one thing that is challenging is that because no gene and no protein works in a vacuum, they all work together. The rest of your genetic background also affects what a mutation in a gene that might cause a disease, how that's going to actually play out, right? And one challenge that they have in this field is that a lot of the way that you kind of determine whether a mutation that you find in somebody's sequence might be important is by looking at whether other people have that have the mutation, that have the other people who don't have the disease have that mutation. And like, what is the frequency of finding that mutation in a normal population? But there's been examples where you see a mutation, you think that it is important because you don't see it very often. You don't see it very often in people who don't have the disease.
Veteran sideline.
And then it turns out that it's just because it's a population difference, right? So in certain genetic backgrounds and ancestry backgrounds, that mutation might be common, and it's not actually causing any problem at all. But you thought it was because all of the things that you're comparing it to are from a different genetic ancestry group. So we are trying to do more to to build out the resources to help interpret variants in a variety of different genetic backgrounds. Because you can't just assume that how it behaves in like a white European background is going to be the same.
Isn't it like Iceland? They have sequence of everyone or something ridiculous.
They have like a very intense genetics program. So they have
and they have the health history of everyone.
Sequence, health history, they've got all kinds of stuff.
But it hasn't necessarily, I mean, it probably won't help outside of that subset of people.
I mean, it's useful for doing things like developing methods to look for things and getting sort of baseline knowledge, but you're right that it is a very specific population with a very specific genetic ancestry. And some of what you are going to learn is not necessarily going to be generalizable to other populations.
But for that subset of population, are you familiar with anything they've discovered or like it's been useful in any?
I don't think I could come up with something right off the top of my head.
Yeah, no worries.
I would say that populations like that, where they are pretty, Iceland is geographically separated, right? And so there's a lot less genetic diversity just because they're all together on this island, right?
Well, they have to consult like a genetic tree for dating purposes, right? Because the subpopulation's so small.
Right, because they could have a higher rate of genetic diseases, that are caused by recessive mutations, where you need two of the bad copy. You need one from the dad and one from the mom to have the disease. So those kinds of things disproportionately affect populations that are sort of separated like that, because those mutations happen on like a founder genetic background, right? And then they just get propagated through breeding. And so you're just more likely to hit that, to find that other allele in that sort of limited in that sort of limited population. So actually the disease that I worked on when I was a technician after I graduated college is one that's prevalent in Ashkenazi Jewish populations. And that made it easier actually to find the gene that caused it, because we had a much less like genetic variation that we had to deal with. You knew when you found the mutation that they were all going to have the same mutation. That's not the case for like breast cancer, right? There are a whole bunch of different mutations in that one gene that cause breast cancer.
I mean, so are those populations better for methodology development? Just because you're narrowing the answer scope you're looking for? Yeah. Methodology development, and also just even answering the questions, right?
Yeah, it does really reduce the search space.
That's fun.
Okay, I remember how to play this game.
You're ready?
I mean, I'm
We're gonna
not
do this?
good at it, but I remember how to play it, so
All
I
right.
am ready.
We're gonna do it.
Okay.
Let's reset. All right.
When do we have to start playing NARC?
I'm gonna say you'll be at about 20 minutes, so we probably have 15 minutes of Dr. Mario time. So I'm gonna let you pick what levels we start at.
Great, we're gonna start easy.
All right. Same level, or should I go higher?
I'll let you start at the same level.
All right,
I
let's
might
do it.
change that.
Let's do it.
Oh, but you can play at a higher speed. Is that?
I don't know if that does you favors at this,
Hmm,
because then they drop faster.
that's true.
It's up to
Why
you.
don't you go on low speed,
Okay.
or should I go on medium? I'll go on medium
Press
speed.
down? Yeah.
I can
Let's do this.
do it.
All right.
Which one am I on?
You're left, I believe.
Yeah. Okay. We're already off to a great start here. Okay, I said I was ready. I wasn't ready. I lied.
Oh man, my wife and I used to play this game so much.
Okay.
But if you guys have questions, I can still look at chat while.
He can
Fine.
do anything when he plays this game.
Oh,
Why don't you
man.
ask him a lot of questions?
Yeah, exactly. Let's talk about photochemistry and spectroscopy. Starting to do a lot of like anisotropy measurements, which is polarized light and seeing how things rotate, the shapes in space. It's really fun stuff.
Of course. Ask him some questions about that.
Exactly. Let's distract. God, I need to play this again. I gotta talk Debbie into doing this more. Ooh, we're gonna set up an elegant one here.
Oh, great. That really, I really love to hear something like that.
It's gonna be pretty. Oh no! Did you see the setup? It was gonna be beautiful. Like the drop and the cross.
Bad for you.
I
Okay.
wanted it.
Okay.
All right.
That's right. We have to play 3, so I have to like suffer through 3 of these before I give you a
No, you're
handicap.
having fun because this game is excellent.
It is an excellent game.
Did you play any of the newer ones? Like, they've released this on subsequent
No,
systems. Oh, that was bad.
no, and I wish I had because I'd be better right now.
Yeah, I don't know how good they are. I've never. This is
Gotta
the
lock
only
in.
version I've played.
I gotta lock in. Oh, I don't like
Good
that
dunks.
sound.
Good dunks.
Yeah, it's when I know something bad is coming. I don't know what I'm doing with that.
Oh, come on. Oh, there we go. All right, there we go.
The problem with this game is like I get like a system of where I'm gonna put things, and then you
Yes.
do something
exactly.
like that,
It's the
which
warfare
ruins
that
my
ruins
system.
everything.
and then I'm just trying to survive, which is what I'm doing right now. Yeah.
The worst part about the drops is they're very slow.
So
And when you lose that advantage, you're in trouble.
also, you're a lot better at like making moves that are going to give you
Yep.
drops for me.
And it was, you can thank my wife for that. She is also very competitive.
Ooh, okay, I got one. And so did
Ooh,
you.
that was a nice one.
Oh, thanks.
That was like a triple.
What am I doing with this?
Oh, this is getting messy.
Yeah. I need to get rid of that.
All right. That will help a little bit.
No, I don't think it's gonna help. It's gonna help you. It's gonna help me.
Anyway, Ask a Scientist Gaming.
Don't forget, you're asking me questions right now.
Your gameplay, expert science. Oh man, we already talked about movies. I don't have anything left to rant about. I could rant about funding because that's been just a
Yeah. So
nightmare.
tell me how things are going in your land of funding.
I mean, we had to wait. You know, well, I feel bad for a junior faculty member. So he's submitting for the NSF career, and he waited 11 months to get his rejection, which means he has 1 month to revise and resubmit
Yeah.
based on those reviews. It means it was probably fundable, but the level just wasn't there, right? So,
So I'm curious about how universities are dealing with this. Because, typically getting grants is part of the tenure,
Yep.
eligibility, right?
Yep.
And at the moment, we're kind of in a place where you not getting a grant isn't because you weren't good enough to get a grant.
Yeah.
It's because there's a lot of other factors necessarily.
Well-
So how are universities responding to that?
Well, so we've had good candidates go up recently that have all had funding, so that hasn't been an issue for our internal decisions. But I'm to the point where I start writing tenure letters, and some of the requests are, did this person actively pursue funding, which is the threshold rather than having funding,
Okay.
which I thought was interesting. But
Yeah.
I mean, it's a reasonable. I mean, the necessity for having 2 federal grants changed to 1 federal grant kind of thing.
Yeah, 2 federal grants, unfortunately, is going to be really hard.
Exactly. Especially since, I mean, getting NSF and NIH funding is rare.
Yeah.
Getting 2 NIH is not common. Same with NSF, that's much less common unless you go to materials engineering and then chemistry as separate grants.
I've just got this whole. Okay, you've got 3 things over there. I have a whole graveyard of
See, for me,
issues over here.
alcohol is performance enhancing in these games because I just relax and just hit the flow state and
It's really great for you.
I promise I'm not talking
No,
shit. This is casual dialogue.
I did this to myself, so I'm not.
That's true. You asked for the challenge. What I look forward to is 4 years from now, when you have played exclusively Dr. Mario for 4 years,
Exactly.
just trade Rocky style.
Exactly. Okay, I'm gonna make you have a handicap now.
All right, let's do it.
So
You tell me what,
do at least 5 more level.
10?
Yeah,
All right.
that seems.
Keep it at medium or
I think that's my problem. I think my problem is much bigger than that. So yeah. No, I think we're all really worried about how everything that's happening in the sort of federal research funding space is going to impact not just the current crop of early career researchers, but
Yeah.
everyone that comes after that.
I mean, so you've experienced several administrations and changes in strategy.
Yep.
Is this something that just bounces back, or is this something there's irreversible damage that has occurred?
It's hard to say. You're right. I have experienced many, not many. How many? But I've been working for the NIH for. Okay. I did that on purpose.
No worries.
That was intentional. I want you to know that.
Do.
And things often change in different ways when an administration changes, but it's never been like this
Yeah.
that I have ever experienced. And so it's really hard actually to know what's going to happen and how we're going to recover from it.
I mean, so there's been infrastructural damage to the agencies, right, in terms of how they do business.
Yeah. Yes.
I mean,
That will be repaired.
okay.
There's also a lot of okay, I stopped paying attention for a second. There's also been a lot of changes just in, I don't know whether this has been happening at NSF, but we are having a lot of different policy changes, changes, all kinds of different changes. And one thing that I would say is that, I don't think anyone would argue that the NIH. Oh god. The problem with having not clearing out the middle is that it
The pile.
ruins everything.
Takes one drop. Yep.
Yeah. Okay. Get back to what I was saying. I think that the changes at the NIH will be solved.
Okay.
And what I was saying was that nobody would ever say that everything that the NIH did the way that they were doing it was perfect. Nobody would ever say that there were not changes that could and should be made. And I don't think anyone has a problem with like thinking about that. The problem is, I think that so many changes are being made so quickly.
Yeah.
And there's how do you know which ones were good? How do you know which ones were not?
Yeah.
The other thing is that some of these changes like really require planning, and typically we plan. Okay, I've done it again. Next, I'm reducing my speed.
All right.
That's what I'm doing. I'm giving myself. Let's just end this right now. We usually do things in a very thoughtful way. One might even call it overly thoughtful.
Yeah.
We get so much feedback from everybody, and we really like think through things before we make changes, and it takes forever sometimes to make changes. Right now, that's not what's happening. And things are happening really quickly. And so one example, I don't know if you remember the like change in indirect rates.
Yeah.
Okay.
Oh my
so,
God, that was so devastating.
yeah, right.
It didn't even happen and it was still devastating.
Right. And that's another example where I think that a lot of people would agree that there is some room to rethink how we approach indirect rates, right? There's definitely, some things that we could do there. But because of the way that our like sort of government academic partnership has evolved over the last several decades, universities rely in many ways on grant funding from the government. And when you change that model, like in a split second, the university is not ready to respond to it.
Yep.
It's okay to change that model, but you have to give everyone enough warning so that they can figure out what they're going to do instead and prepare for that. And so that's kind of where we've been operating differently than we normally would.
And so the other thing we've seen, at least on the NSF front, is a reprioritization of goals, right? In terms of like AI, machine learning, quantum all of a sudden became the most important thing.
Yeah.
I mean, have you guys seen that as well? I mean, we've just learned that vaccines cause autism, so obviously that has implications, right?
Vaccines do not cause autism.
That was a sarcastic
right.
joke, ladies and gentlemen, for people not familiar.
FYI.
Yeah, exactly. I apologize.
But so we have had changes in priorities, and you've probably heard about some of these things, and these are also not things that only impact the NIH. It is true in many different areas, but about really de-emphasizing DEI.
Yeah.
Now, you could argue that we don't all have the same definition of what DEI is and how that relates to research, and we may have overcompensated. So that's really the main thing, is that there has been some shift in the kinds of research that we will fund. Oh, okay, fine.
And so it changed, like in terms of research studies, you can't do like targeted studies for women or minorities has it gone to that extreme?
There's a lot more scrutiny about that. Now, I will say that where I am, I'm kind of insulated from that in many ways, because, wow, okay, I've heard that sound a lot.
Sorry.
That means you're killing me right now. So the kind of research that my division supports doesn't that doesn't really come up that often because we're like developing sequencing technologies, right? And that's doesn't have anything that anyone objects to.
Yeah.
And I also would say that those kinds of changes in priority have happened before. And to some level, you know, the NIH leadership does have that's it within their purview to set different priorities. But again, it's like the way that it was implemented.
Yeah.
It's okay to say going forward, we're not going to fund this kind of stuff anymore, and we're going to fund more of these other kinds of things.
But to try to pull back funding.
But we've never canceled them.
Yeah.
In the middle, right?
That's crazy.
And that was a big difference, and that's very disruptive to people.
Yeah. And so, interestingly, a lot of this affected NSF grants in terms of we have the broader impact statements.
Right.
NIH, you guys don't have that. Was there ever discussion at any point of including that in like a criteria?
I've never heard that, although I think that it would be good because my understanding of the broader impacts, and you please correct me if I'm wrong, is it's basically about public outreach, right?
Yeah, public outreach and like, are there actual implications to your work?
And
is it fundamental, or could it do something useful?
I would say that something that kind of got us in the position that we are in right now is that we've clearly not done a good job of communicating to the public the impact of the research that we do at all levels, including basic research. We haven't done a good job of communicating to the public how science works and why our understanding changes as we continue to like do experiments and learn more about things. It isn't because we were wrong, but it's because we learn more. And as we learn more, just like the variance, like we learn more about variance over time, and then we're able to reinterpret things that we'd already identified, but we didn't understand. And now we have more data and we can interpret them now. That's how science works as well. And it's very clear that we have not done a good job of explaining that. And so now we're in a position where a lot of the public has become skeptical of what we're doing because we're doing science in exactly the way that it happens, right?
Yeah.
And that, yeah. So I think the broader impacts, like, that's important. I think we do need to learn how to communicate better.
I mean, I think one advantage the like NIH type research has is that it's much more tangible in terms of I'm going to cure cancer, or I'm trying to understand this to cure cancer. Whereas, you know, us on the, you know, fundamental,
I agree.
like cis-trans isomerization, who cares, right?
Right.
Like, but eventually it might be useful for MRI imaging or whatever it might be, the long-term implications.
And to be clear, we actually also do a lot of research like that. That is very basic. And research like that needs to happen because you don't always know when you're doing it what is good. Oh, thanks.
Yeah, what's useful.
What it's gonna turn into, right? I think that there are a lot of examples of fairly basic research that turned into something down the road that has become quite important, like the GLP-1 drugs, right? Like,
oh yeah, that's a fun example.
Yeah, like that is like from a, was it identified in a Gila monster?
I don't know the origin.
Like, but anyway, it was the whole thing was they were not looking for drug. They were not looking for a drug. Like the understanding of that, you know, pathway came from very basic research. And then, you know, as we learn more, we're able to think about how that might apply to human health. But if you stop doing the basic research, like you're never going to get to these new drugs because you don't have anything to start with anymore.
Yeah.
And I hear a lot of discussion like, oh, well, that will happen in companies, but it doesn't.
It doesn't.
Because companies are focused on making a profit, right? They actually rely on people in universities to do that sort of initial foundational research.
Well, not,
And then they figure out what is promising and they go forward with it.
I want to emphasize their profit is quarterly based. It's not decades based. They don't think long term in research.
Yeah.
That's fair. I am digging out. I'm trying.
Wow. Okay, that's great.
Oh no.
Well, it's not.
I love the grind though. It's fine. It's fine.
I'm pretty like. I've got some problems right now.
It's a question of who can dig out of their mess
Yeah,
first.
yeah. Okay.
Who can dump the most shit on the other person while they try?
I'm not gonna. Oh, what are you doing? Stop.
Surviving.
Oh, okay. I just need to unearth some of these. Like, I can't get to this.
Oh man, it's 10:45. We've already played it for like 20 minutes.
Oh no, we have to play the part.
Yeah, we do.
It's gonna take me more than 15. Okay, we need to stop this.
Oh come on, we can finish this round.
Okay. Oh my God.
Yeah, we got this. I'm sorry, I'm keeping you longer. I apologize. But man, it is fun to be playing this game again. I've completely ignored all the things. If you guys have questions, put them in chat. We'll get to them as Lisa plays NARC. But for now, we are playing Dr. Mario, which is an all-time great. If you guys have never played this game, you definitely should.
If you've never played this game, don't play it against him.
It's an underappreciated gem of the Nintendo gaming age.
I made it. I should have done something different there.
Oh, what am I gonna do with you, yellow? Trapped.
Oh no, I've made a huge mistake.
Get my booze
I
out of
could
here.
have like solved this problem, and I.
Oh, idiot.
Oh
Idiot.
no.
All right. Okay, we're doing it sideways. That was a good one. That was a fun battle. It was speeding up hard. All right. All right. We got to get to NARC.
Yeah, this
All
is
right.
going to be another.
Do you want to see the clock or do you not want to see the clock?
No, I don't. No, I
Okay.
don't.
Okay. I will just put the clock somewhere. Oh no, it's updating. Update clock. There it is. All right. Does that show up on screen? Awesome. I will put it over here so you don't have
Yeah,
to
I
look
don't-
at it.
No, I will say, yes, I've played this game before
And it's
on this
been
very stream,
4 years and you played it once.
right? And I meant to watch it so that I could remind myself how to play it, and I forgot.
Oh man, I should have sent you since the last
No.
time I generated a YouTube tutorial on how to
Oh.
beat this fast. But for now, before you press start, I don't know if I had this last time you were here. This is the original like box that came in, and you're going to learn the lore because the back of the box
Okay.
has the story. So, NARC, no one had the guts until now. You are Max Force. Your mission: bust Mr. Big and destroy his dreaded criminal empire. Seize all contraband, stolen money, illegal weapons. Use rocket bombs, high-powered machine guns. Apprehend all suspects. Protect the innocent. Punish the guilty. Stop at nothing.
Okay.
So go ahead and press start. The goal of the game is to walk right and find doors as quickly as possible and killing people doesn't have an advantage, but while you walk right, you can practice jumping and shooting rockets.
Yeah,
That's the skill set that becomes very relevant later. So just hold right. Yeah, exactly. As long as you get good at that, you will be fine at the end bosses. Jump, rocket, Jump
I
rocket,
love
jump
how
rocket.
when you kill them, they just like explode into
man,
like a
see
bunch
that
of
door right
body
there?
parts.
Exactly, and that was one of the reasons. This was one of the early contributors. This, along with Mortal Kombat, for the like game rating. This was what Nancy Reagan was rallying against, and music and video games being too violent and whatnot.
Oh, wait, do I need to go in there?
You need to get that red card.
Oh. Well, how do I get it?
Yeah, go right.
Oh, keep going.
There'll be a door at the end. But yeah, if you guys have any questions, Lisa's happy to answer them. We're talking about, well, I mean, we've journeyed through many topics before. But yeah, we can talk about genetics, we can talk about epigenetics, we can talk about genomics, NIH, life as a program officer. Speaking of life, so you did the transition from working remotely for a long period of time to all of a sudden going back to the office.
Yeah.
What was the weirdest? What was the, what shocked you the most?
Driving.
I completely forgot how to drive.
One thing
First commuting, the better, like, because that's time.
Well, yeah, so like it was very hard to get used to like having to get my lunch. I bring my own coffee in, I bring water in, get my like everything. And get out the door and exercise and like you know, figuring out how to fit all of that in the morning was challenging. But the biggest thing is that I just really was blissfully unaware of like what rush hour traffic was like.
That's fair. All right,
Oh,
so
I
here
need
you
a
gotta
blue
shoot-
safe card.
You have to shoot those guys with bullets at close range. So not rockets anymore. So it's
Oh.
going to be hold bullets, go this way.
How do I do bullets?
Hold A instead of tapping A. Yeah, there
Oh.
you go. So turn around and shoot the guy in black.
Oh, and now I'm getting eaten by a
And
dog.
if you hold both buttons down, you can shoot the dog. Yeah, just hold them both. Yep. Hold, yeah, there you go.
Yeah.
Okay,
Okay.
shoot those guys at reasonable close range. Bullets holding A.
Yeah.
And this is a random number. There you go, blue card.
Okay.
All right, through the door. Or
Great.
to the key slot, then the door.
Okay.
There we go. All right.
Yeah. So
Rush hour.
yeah, I really didn't realize what rush
So-
hour traffic was
so
like.
are you-
And we live in Tallahassee. Let's
Yeah.
just be clear.
Yeah. Rush hour loosely defined.
Right.
But that's, I mean, so you're heading to campus, but you're on a fixed 9 to 5?
Yeah, pretty much.
No,
Yeah.
Okay. But is that window, you define that, or that's?
I mean, people can work. You just work 8 hours, right? And
Mhm.
people can work other times, but that is the schedule that I work. That's the schedule that I would say probably most of my colleagues work.
Oh, that makes
Oh,
sense.
now I get
Jump
in
in
this
the car.
I
Yep.
don't-
Tap B, and then stay on the bottom.
Yeah, I remember this.
And then
not
you'll run
going
into
well.
something. I'm impressed you remember any of this game. Oh, Lori redeemed NARC drink and drive, so cheers.
Great.
Lori, always a pleasure. Thank you for catching this appropriate moment in the NARC
It's okay. I'm not at my car yet, so
Oh man, that's okay. You've been commuting to work. You've been training this for some time.
yeah. So that. Oh, but I got a new car,
Oh, nice.
so that made driving a lot more enjoyable.
I drove so many junkers as a kid, and now I have like cars that run well. And
Yeah.
no, it's pretty satisfying.
Yeah, it really is.
Anyone that talks shit about minivans has not owned a minivan. They're outstanding.
Okay.
No.
Agree to disagree.
Oh man, with a 9 and a 6-year-old, they are amazing. But yeah, anything else? I mean, you went from phone to like Zoom and Skype.
Yeah, so actually that transition happened a little bit before COVID. Having Zoom changed my life as a remote person.
Yeah.
First of all, I would say that I am remote because I had to move here and I needed a job, right? And I wanted to keep my job. I'm not remote because like that's the path I would have chosen for myself necessarily.
Yeah.
I like telework. I think everyone likes teleworking to some degree, and I think that like that's obviously good. But
Okay, stay on the bottom, and then go up to the top white line.
When?
to this right now, and then you'll run into something, but that's fine.
Okay. So I don't remember what I was saying. Give me a refresher.
You didn't choose.
Oh yeah, I didn't choose. I'm
Remote.
very extroverted.
Yeah, you didn't
do-
want it because you could wear pajamas every day.
Right.
It was strategic.
Yeah. If I had my way, I would have stayed where I was. I would have kept going into the office with my colleagues. But I didn't, right? We had to move here. We moved here for my spouse's job. And I was it doesn't make sense to lose staff for that reason when you don't have to. Okay, I have to jump
So
in.
stay on the bottom of the screen all the way to the end. And then, yeah, that's the end of the level.
Okay, good. So anyway, yeah, it I'm glad I wasn't
Gentler.
right. I'm glad I was able to do it because I like my job, and I'm glad I was able to keep it when we moved here, but I didn't love working from home.
That's fair.
So even though it's ridiculous that I go into an office where I don't work with anybody that does anything to do with anything that I do, it is nice that, okay, clown.
With the knife.
It is nice to be around people, and that part of it I like.
Has the being around people hurt your productivity?
Not really. Oh,
Okay, keep going. No, keep
Okay.
going. That door right there.
this one?
Yep.
Is it cheating if you're...
No, I walk everyone through this. I
Okay.
don't want you to. We don't need to spend a half hour. Grab that card, press up, but when you go through the door, don't keep pressing
Right.
up.
Yep.
And left. Yep. That's nice. Sometimes people walk in again and then have to do the loop. Your pace is excellent. You're doing well.
Okay, great.
got this.
Well, I haven't gotten to the
Mr. Big.
big scary head man
Yep.
yet. Yeah, so I wouldn't say that it's impacted my productivity in that I'm like
Through the door, and you're gonna go left.
Right.
You're gonna
Oh,
shoot
I
these Rambo
remember
guys
these.
with
I remember
bullets.
these trees.
So,
bullets are
Yep, holding A.
this.
But you want them closer range
Oh.
because this game's dumb. All right, turn around and go back the other way.
This game is really dumb.
It is.
This game.
Who picks this game out? Does it go left? You want the Rambo guys and shoot them when they're close.
Okay.
Yep, just hold it down. Keep going
Oh.
left. He'll come back.
Oh, okay.
There you go.
Oh,
green card.
okay,
Perfect.
great. Okay,
Perfect.
great. Let's get out of this area. Okay. Yeah. So, but it is a very interesting, like, working environment there.
Mhm.
It's very different from my office environment,
Yeah.
right? Like where I work, I do go to the office sometimes. And we have offices and everyone is just in them, and we spend a lot of time on Zoom anyway, even when you're in the office, because we're talking to researchers, we're talking to people in our research consortium, like we're on Zoom a lot. And people are just in their offices doing that. This office is very different in that it's open.
All right, yep.
Oh, they're all they all have to talk to each other and communicate. I was worried that they were going to be annoyed with me being on Zoom constantly behind them. I think that they are. If you are, guys, don't tell me.
Yeah, we'll play it
I don't.
naive.
I don't think that they
Ignorance
are.
is bliss.
But I think it's because, like, it is a very sort of, it's already an, now how do I get that
You
red
have that
safe
card.
card?
You have
Oh,
it.
I do?
Yep. Just go directly.
Okay. It's already an open environment where there's already people talking all the time. So I think they've all evolved protection mechanisms,
Yeah.
which include listening to headphones if they need to
Yeah.
concentrate.
Noise canceling headphones are the best.
Yeah. What I would say that it has changed is that I don't work more than 8 hours typically, unless I really have to. And I used to do that
Oh, interesting.
all the
I
time,
guess
right?
it's because it's
I
always
have that
there.
one too, right?
Yep.
Okay. And so that's what I would say is that the government gets less out of me than they used to, just because I have to commute home now.
Oh. But the social aspect has been better for you on that front.
Yeah, for sure.
That makes sense. So my wife has been working remotely since we were in grad school. So it's been
Oh, wow.
16 years.
Oh, this guy?
Not yet.
No? Okay.
Keep going right. You got to fight a wheelchair guy first.
I really don't like the dogs.
Hit both buttons.
I know.
And, just hold
I
them
know
down.
you said that, but like in practice, that's not going as well.
Yeah, just hold them and like weave back. Yeah, there you go.
But there's more dogs.
But yeah, she would sometimes fly back to USC to join people in the office, and she was surprised by how much they talked to each other and how annoyed she was by it.
Yeah.
But she's also mildly
do I have
introvert.
to kill
Yep,
somebody?
Wheelchair guy. Okay, so
Oh.
that's one. You have to do that. So keep going right. Kill everything on screen. He'll come back. You have to do that twice more.
Dogs.
Oh, there you go. Nice. Perfect. Yeah, but when the wheelchair guy comes back, rockets. But these guys just kill, however. Oh, there he is. That is a good strategy, believe it or not.
Yeah, I mean, I would say that for sure there are things that are better done in the office. And that's why I travel to the office sometimes. And I go to those things in person. I do agree that there is some
Stay over that way.
some things that work better that way.
And so these are like meetings, like the major meetings of the consortium, or?
Those I definitely go to in person.
Yep, there you go. Whoop, that's number 2.
Okay,
All right.
is that it?
Nope, one more.
Oh,
It's all right. You're having fun. This is a great game.
right.
What's your guess on the world record for this one?
Oh, I actually, it was very short, like 9 minutes.
9 minutes and 3 seconds. So, last time you were on, I had the world record, then I lost it. December, I
right.
got the world record back.
Oh, wow.
And so it was fairly recent. Not only that, my colleague Eugene DePrince joined me on 2-player. And
How
we
many
hold the
tens
2-player
of people
world
play
record.
this game
Oh
now?
man, there's at least 15 people on the
Really?
leaderboard.
Is there-
So of those 15, I'm on top. But yeah, I was very proud. Eugene and I got the 2-player speedrun record as well.
When you
This is-
play this 2-player, what do you do?
It's
Is
just
it...
2 players doing this exactly.
Oh, I got
stay,
him.
stay right. Nope, left.
Oh.
Stay there. He's gonna drop
Oh.
a card, and if you lost that card off screen, it would have freeze you in the game.
Okay, I
So
want that.
yeah. All right. No, your pace is great. All right. Now it is time
Okay.
for Mr. Big. Is there any
Okay.
questions I should have asked you that I didn't? I think we got through early career research stuff.
That
we
was
didn't really...
one I thought was interesting about the identical twins.
We can pop that up quick.
Let's do it.
How many people are still around? Because we're after 11 o'clock. All
Yeah.
right. Top of the
Oh.
screen, jump rockets. Yep.
Right.
Yep. So get some distance. All right. I'm going to put up a last question of the evening. We're going to ask a question about... All right. So he's off screen. Jump. Oh, too hard. Oh, close. That was a really good rocket. All right, we're going to put up a prediction. Last prediction of the night, if you guys are still around. Answer this question. Identical twins have identical DNA, true or false? Or yes or no? I guess I put yes
Yeah.
or no. All right. Anything you want to add to this question, or is that, I mean, it's pretty straightforward. Identical twins have identical DNA.
All the time. All the time.
Okay. Identical twins have identical DNA all the time. True or false, or yes or no. Put your answers in there now while Lisa takes on Mr. Big.
Now, what? These are his tongue?
Yes,
Okay.
there's tongues.
Right.
That's why stay on the top. You'll avoid most of the tongues. I think you're pushing it a little
I
too
think
hard.
so, too.
Gentle. Oh, that was a good one. The jump was good.
Yeah, the shooting less
Yeah,
good.
It was close though. It's like jump, then shot. A little earlier on the shoot. Very close. All right, put your answers in now. Identical twins equals identical DNA, yes or no? Answer your question, bet your internet points accordingly. Also, if you guys have suggestions on anyone we should raid, education or related, there aren't a lot of streamers on Wednesday night that I know, so if you guys have suggestions, let's do it. Let's find some new people. I'm happy to take suggestions of any kind while Lisa fights Mr. Big. What's more stressful, this or surgery?
Oh, this.
Really?
Because I
The
can't
clock?
give up.
Oh, that's true. there's no options.
I have to, at
That's so
some point,
close.
beat this guy.
That's so close, though.
It's the weird jumping.
Yep. So
Those
just
of you who are not playing this
press
game,
them lighter. Yeah, try
there's
to.
only 2 buttons, but they do like 4 different things,
Yep.
and it depends on how you, like, how gently or quickly
Yep.
you press them.
So you have basically 6 frames to get a jump button off, which means you have 1/10 of
Oh.
a second to push that button.
So if you
Yeah,
press
go a little higher.
jump too hard,
Yep.
you're not jumping.
You squat.
If you press shoot too hard, you're not shooting or something.
Yeah, you shoot bullets
So
to
that's-
rockets.
There's like a lot of nuance to this.
I
that,
strongly encourage any long-term viewer of this stream to just bust out NARC emulator and just give it a try, and watch my tutorial on YouTube.
yeah, so that you can understand why I look like I'm having a seizure right now.
That's what makes it fun,
And
right?
like why I cannot seem to do the most basic video game things like jump and shoot. It's because
It's hard. Ooh,
have I
that's
hit him
close.
at all?
So you got to hit him in the hat.
I
The,
know,
the
I'm
first one has to be
trying.
that, otherwise the rest don't like hurt him for unknown reasons. So this is what the war on drugs looks like. You remember the 80s?
I do.
Very similar.
Yes. Kinds
Oh,
of
man,
like
it's-
big
speak-
heads
Speaking of which,
with hats.
Lisa might appreciate it.
I can't look at it because I'm very busy.
Grew up in the Midwest
I did.
in the 80s.
Oh, I did probably have one. Of
I have to show anyone my age, I have to show them my DARE certificate. My mom sent me a bunch of crap like 6 months ago, and this was in the pile. So I officially have my DARE certificate. And obviously, I have not done drugs since then.
How many people in this streaming thing spend 3 times as long in Mr. Big as they did
On the
in
rest of the
any
game,
other part
this,
of this?
this is the rate limiting step. But your pace is still good.
It
It just takes one good rocket to the hat. Oh, that one was close.
close so many times.
I think it's like dot dot. I think the like calm like it's hard because your second press you always want to push hard because you're panicking on the switch between them. Because you're getting the jump, but the rockets aren't coming out. So I think you're pressing it too hard. Oh, that was a close one.
I'm barely getting the jump,
Bad.
and I'm definitely not getting the rocket.
I look forward to next
So the
time.
rocket, I have to press harder?
No, gentler. If
Oh.
you see bullets come out, it's because you pressed it too long. I look forward to the stream 4 years from now when Lisa comes back, just elite at Dr. Mario and NARC. Like, all you've done is those 2 games.
I really should have
Oh, man.
studied this
Hey,
a little
do
bit
you
more.
play Dr. Mario against your kids?
No, I
Oh,
don't even
man.
have it.
You can probably get it for the Switch. It might be on the
Probably.
NES emulator, actually.
I do have an emulator. My brother made one. I can't remember how to turn it off.
No worries.
It's very complicated.
Yeah. That's unfortunate. But yeah, I think the Switch, there's a built-in NES emulator, at least for the Switch 1 there was. I haven't checked on the Switch 2. But I'm pretty
Yeah,
sure
there probably is.
I'm pretty sure Dr. Mario is on there. And I should play more. I miss it. I miss it. But yeah, last minute suggestions on people we should raid and or questions you guys might have. Let's do it as we close out the night. Winning the war on drugs. Everyone's happy.
I'm not winning the war on drugs.
So one last thing I wanted to ask you about is program officers, at least some of the ones I've talked to at like DOE and NSF,
Yeah.
like they can't accept, coffee from someone, right? Like what are the restrictions? I mean, any that stand out to you that people might not know?
I mean, there is like a dollar amount. I want to say it's like $25, $15. I don't accept anything from anyone. I brought my own wine here tonight.
Yeah.
It's easier to not accept anything.
Yeah.
Yeah, because it's very important that we are visibly impartial, right?
Mm-hmm, mm-hmm. And that's just anyone, or it's people that might be in the funding sphere?
I think that are in decision-making sort of position. Oh my God, this is gonna go on forever.
Man, that's crazy. And so how does it, this might be a sour topic, but when looking at the upper branches of our government where those rules don't apply,
I do
that's crazy,
frequently
right?
notice that they don't seem to have taken the same ethics trainings that I have taken.
What does that look like? Is that like an online module? And
We've got all kinds.
and has it changed over the years, or is it pretty consistent?
No, it's been pretty.
The same rules.
This is the most annoying game in the entire world.
Absolutely. I agree with you. Who would make their guests play this? But last minute questions, last minute suggestions on who we should raid. I'm going to take a look who's available. Because I don't know who's on right now, but science and technology, always good. Building PCs. Some shark streams, earthquake streams. There are so many random streams out there.
I can't even shoot the rockets anymore. Like, I keep shooting bullets, I think.
Old Man Felonious, welcome back to the stream. They have taken the same ethics training I have. Same training, but different results.
Yeah.
I mean, so what would happen if somebody found out you had accepted money? Like, what's the repercussion and who enforces that?
Actually, I don't know because it's never happened. I mean, not that it's never happened to anyone. I've never... Now, okay, I'm shooting the bullets.
So try the A,
But...
Yeah, it's gentler.
Ah.
There you go. Yep, you got to get
Okay.
both those taps roughly as light.
The problem is I'm so...
You could try the piano strategy, like put it down on the table and... Old Man Felonious late to the game tonight. No worries, you're here for the best game of the evening. NARC,
Yeah.
we're going to win the war on drugs. Our guest tonight, Lisa Chadwick. She's a associate director at the NIH in the program. What's the name of your
Division of Genome Sciences at
never gonna
National
remember.
Human Genome Research Institute.
So her expertise is in genomics, genetics, epigenetics. If you have any last minute questions, he's happy to answer them while trying to win the war on drugs and fight this giant head with rockets. But yeah, we've journeyed through a lot of topics tonight. Oh, there it is. Okay, now to the face. Just no more jumps. Let him get a little closer and rocket. All right,
Whoa!
run away. And you're going to use bullets, and you're going to want to be between these two lines shooting bullets at him.
Between
Yep.
those two lines.
Yep, these two black lines. Just whatever Nintendo rules. But yeah, get some distance away from him. He no longer shoots anything, so that's nice. But that was good. I like it. Yep, get some distance. Get him off screen. Turn around and just hold bullets. And then... There's two of them. All right. Now you're just going to want to move up and down just a little bit. But yeah, we journeyed through many topics tonight. If anyone has suggestions on who we should raid, we're happy to take a look as we close out the evening.
Obviously, my expertise is not in war on drugs.
I mean, also this controller is a blast from the past. The crippler. People might complain about the WiiU deck, but it is
Oh yeah, this
more
really gets
ergonomic
your
than that.
thumb.
Yep.
My thumb is numb. Okay, I would like to get away from you. I need you to leave me alone.
It's kind of sad when I like speedrun Mario, my hands like hurt afterwards.
Now,
Yeah, you have plenty of distance that way.
Yeah, if I can get away from him.
And then just turn around and stand there and bounce up and down just a little bit. That was good. Now run away. Try that again. Down here lower. Yep. Eventually one of them's gonna go. I promise.
Sure.
It is beatable. In fact, you've beaten this
I've done it before.
Exactly. This will be your second time. Veteran.
I remember it being annoying last time too.
You need to challenge your daughter to this game.
Oh,
Turn around and shoot. Done. Go right. Go right. It's done. Oh man. Speed strats. But you feel the accomplishment.
Was this like a game that you could play in an arcade?
It was. It was an upright arcade, and that's when they translated it to the Nintendo.
People paid money to play this
Well,
game?
so it wasn't this version. The actual arcade one is much better than this game. In fact, next time you're on, we could play it on the GameCube.
That's not a high bar.
No, it's
I don't need
much
this
prettier.
goal, do I? I can just
No,
run.
you just
Okay.
keep going. Find the door.
I'm not motivated by money.
That's good. Except for that one. All right, you did it. 26 minutes, 39 seconds.
That's not really better than last time, is it?
It's so I think you got to the Mr. Big faster this time.
Yeah.
But you were slower on the headshot.
Yep.
Next time. But you still get to put your initials in
Oh, wow.
with the mutated finger, whatever that might be. All right, ladies and gentlemen, we're going to close out the night. Lisa and I stole extra time because we played Dr. Mario, which was a lot of fun. So we've run 15 minutes over. I apologize. It is later than we usually are, but totally worth it. I really enjoyed it. Blast from the past. But anyway, Lisa, thank you for joining us again. You weren't so off put by NARC last time. Maybe this time we'll see, but I
I'll
really
build up
appreciate
that callus on my thumb
Exactly.
for next time.
I really appreciate you coming back and joining us. It has been a pleasure talking through things. Any parting words for the audience?
No, I really enjoyed being here and talking to people about this, and I hope you'll have me back another time.
Oh man, it'll be 4 years from now. But actually, the list has gotten quite long, so I cycle through people. But it was a pleasure. I really enjoyed talking, really enjoyed playing. It was a lot of fun. We journeyed through Super Mario 3D World, Surgery Game, Trauma Center.
Yeah, Trauma Center.
Second Opinion, which was a lot of fun. If you want anime drama while doing surgery with a laser pointer, basically, that is the game for you. We also did some Dr. Mario, which again, this is only the second time I've played against a guest, so Lisa, it's a pleasure as usual. And then we closed the night with NARC, 26 minutes and 39 seconds. So thank you all for joining us. Any last minute suggestions on people we should raid, put those in chat. If not, it's been a pleasure. We will have a guest. Actually, it's not in 2 weeks. It's actually next week. Our guest will be Dr. Dawn Carr. She's in the Department of Sociology, and she studies old people, health and aging, elderly care. I mean, it's a huge contingent, a huge push, part of FSU Health. So I look forward. It's been 4 years since she's been on as well. So it'll be fun to have Dawn back on. I don't know what she's playing yet. We're still going to figure out games and everything, but it'll be next week. And then after that, we'll be every 2 weeks through the fall semester. I'll update the schedule sometime soon. I just got to fill in a few additional slots. But with that, I mean, I think that's everything.
Okay. Say no to drugs, everyone.
Yeah, exactly. Just say no. Fun factoid. On the arcade version, that license plate says "Say no or die."
Oh.
And then Nintendo made it gentler by just say no. So, fun fact for everyone. All right, stick around. We'll probably raid someone, but it's a pleasure. Thank you guys. The questions are always fun. The discussion is always outstanding. We really love that you spend this time with us. So, thank you all. Hopefully, we'll see you next week.